pharmaceutical market. And the $4.5 billion US in 2020 is expected to go to 18 billion by 2025. And the post-COVID pandemic era has further increased this demand, where there is a need for prevention and therapy. So, in contrast to pharmaceuticals, we know nutraceuticals are much more friendly, less expensive, safer, and preventive as well. So in the current population demographics, where we are seeing old age population increasing exponentially, and stressed populations and healthcare system are getting structurally stressed, there is a compelling need to promote nutraceuticals and nutraceutical research. So especially the attributes that are known as not disease, such as aging, stress, do not need drugs, but if we can intervene then with nutraceuticals, we can decrease a lot of burden on the drug development and healthcare system. So just a quick note to say that in vitro cell culture system have been providing a powerful research tool, and we know that many landmark discoveries have been made by using this cell culture in vitro system and then in vivo working on rodents. So I've been working on the molecular mechanism of stress, aging, and cancer all these years. And in my research as well, using cell culture, Renu and I have discovered two proteins named Mortlin in 1993 and the CAF in 2000. And both of these play a major role in stress and cancer, and hence we were looking for inhibitors. That's how we met nutraceuticals. And there are several natural resources my lab has been working on, and we are best known by our research on ashwagandha. So we have shown through our experiments that ashwagandha leaves have anti-cancer activity, and it belongs to more than one component. These components segregate in alcoholic and water extract. Consuming crude extract or just a leaf powder is better, as it would give you more prevention, multi-molecular prevention. So we also showed that mechanism of selective cancer cell killing activity, not only that, but also showed that there is a side effect from the components of ashwagandha, which protects normal cells. This is in sharp contrast to chemotherapeutic drugs that are extremely toxic to normal cells. We also showed that ashwagandha vidanolites possess anti-COVID activity. The receptors TRPS2 and ACE2 both are decreased in response to ashwagandha, and hence inhibition of infection was predicted. Now, in real PCR antiviral assays, ashwagandha caused 90% of inhibition, whereas remdesivir was 99.7%, as this experiment was done in collaboration with Regional Center for Biotechnology, India. And this was reported in all the news as early as 2020, May, when pandemic had started. Now, we are AIST India International Lab with several sister labs in India, including IIT Delhi, IIT Guwahati, Guru Nanak Dev University, Manipal University, Sikkim University, and so on. So we host a spectrum of education and training programs. Our Cafe Plus, and workshops are popular. All information is available if you click DiLab in Google's. Now, we as the DiLab team have been associated with INCD since its conception. We have been holding sessions called Picnic. This is the one in Goa. In second, I think it's the second INCD. This one in Rishikesh. Puja and Aarti Puja, we have a strong memory. Our members are sitting just right in front in the Ganga Puja. And this one is in Guwahati, which was organized by Professor Ajay, and such a lovely place and cultural experience. And here we are today at the 5th INCD. And in today's session, there are 6 speakers from different INCDs, different DiLabs. So without taking much time, I would like to invite the first speaker, Dr. Renu Wadhwa. She had her, so while you can set up a computer, I can just introduce her first. So, yes. Okay. Okay.
Now I would like to call on the same chairperson of the conference, Professor D.V. Agarwal. So, Professor D.V. Agarwal, founding director, his current application in the Informative Research Centers and California, USA. His area of research is in cytoskeleton research, molecular therapeutics, cell and molecular growth regulations. He has done his PhD in the biochemistry from the University of California, Berkeley. Renson Home designation professors of the Experimental Therapeutics and the Cancer Research Institute from the Biochemistry and the Immunology Department. He has invited reviews and the book chapters. He has introvascular and the introventors on the over 33 patents. He has more than 13,000 citations. He has the chief of the cytoskeleton research section in the Department of the Experimental Therapeutics at the University of the Texas MD Anderson Center in the Texas. We all welcome you, sir. Thank you. Now we have our third chairperson for the day is Professor Anju Katyal. Professor Anju Katyal is a designated professor in the Dr. B.R. Ambedkar Center for the Biomedical Research, North Campus, University of Delhi, India. Her area of interest in immunopathogenesis, mechanism of disease, cerebral malaria, cerebral ischemia, and the cerebral hypoxia. She had done her PhD in the parasite immunology from the Postgraduate Institute of the Medical Education and the Research, Chandigarh, in 1983. She is a visiting scientist in Department of Biology, Georgia State University, Georgia, Atlanta, in 2012. She is invited to deliver a lecture at a symposium like the legacy of the nitrous oxide to commemorate the 100 years of nitric oxide currently. She is a member secretary in the Institute for Animal Ethics Committee from 2002 till 2013. She is a reviewer member of the scientific journals. She has achieved a DST Young Scientist Award also. She has 59 international research publications and more than 1,000 citations. Thank you, ma'am, for coming here. We all welcome you. I just request all the chairpersons to preside these sessions. Start.
Thank you.
Yeah. Sure. Now you can go.
Okay, so let me invite the first speaker, Dr. Renu Wadhwa. She had her first PhD from Guru Nanak Dev University and second from University of Tsukuba, Japan. First postdoc from University of Newcastle upon Tyne and some time in CCMB, Hyderabad, and then postdoc in Japan. And then she moved to industry for 7 years and then came back to National Institute of Advanced Industrial Science and Technology, AIST, where she is a prime senior research scientist and also heading the lab. We call it as International Laboratory Lab. And with this, she has been awarded with several fellowships from international and publications. So with these few words, I would like to invite Renu. All yours now. Okay.
Am I sharing screen? Am I sharing screen? Okay. Right. No, it's not the right screen. We are not sharing the right screen. Okay. If you allow me to share. Right. This is the right. Okay. Good morning, everyone. I'm First of all, thank you organizers, Alokji and the team for allowing us to have this picnic here, Series 43. And the title of my presentation is not really a very pleasant one. We are talking about stress and aging, but I guess if we deal with our fear and our scares, it's better off later on, right? So I'd like to start with the contents of my talk, and to begin with, just to remind you that these two phenomena, we are talking about aging and stress, they are indispensable. There is rather no escape from these, for we need to learn how to deal with this in a pleasant and a nicer way, non-toxic way, and that's this whole meeting is about. So I like to remind you that the cell culture system provides a very powerful way to understand the mechanisms of stress and aging, and just like us, we age, we stress. Our cells in culture also show stress and aging in terms of their whole physiology, metabolism changes. And we know that these areas are the priority research for the reason that the demographics of aging and stress, they are moving towards about scary thresholds. The healthcare systems will be on alert. They are already on it. So we need to find a nicer and cooler ways of dealing with these very indispensable phenomena. Now, reminding you, my lab also has been working on stress, aging, and cancer together with these. Just to remind you here that cancer is something which is very closely related with age. As you go beyond 50, 60 years of age, you see the increased incidence of cancer. Similarly, in the cell culture system, as the cells age, they have more probability of getting cancerous. Now, this is just for people who are not doing cell culture research. This is how the cells look like. The normal cells, they will age and look like aging here, you know, in the culture dish. And the cancer cells, on the other hand, they can go on forever. Using this system, over last, more than 3 decades, our lab, Sunil and myself, we've been working in Japan on these, research problems. We have been able to discover a protein first in 1993. We named it as Morlin, and the second protein discovered by our group is called CARF, and both of these are now known to enhance carcinogenesis. They play a role in human carcinogenesis, and that's the reason we, as Sunil also said, we've been looking for inhibitors of Morlin and CARF. Just a brief CV of Morlin. From 1993 onwards, we've been able to show several, roles that Morlin play at cellular, at the animal levels. I just made this little CV slide for its working, professions, likes and dislikes. So I'm not elaborate my talk on Morlin today, but just as a summary slide, what we understand about Morlin's role during stress, aging, and cancer is right here. It's a stress family protein, HSP70 family protein. As we age or as cells age, Morlin decrease. And this decrease is itself is a stress for the cells. And the cells try to adapt to it by putting Morlin on the higher levels, but they forget where to stop. So Morlin goes beyond threshold levels, bring the cells to cancerous phenotype. So that's all we know in 30 years of research, and each of these steps is published as a molecular mechanism in several papers. So today I'm going to elaborate on the second protein, CARF, and this was cloned as a binding partner of a tumor suppressor protein called ARF by yeast-2 hybrid screening. Early on, we knew that if we put extra copies of CARF in cells, they give stronger growth arrest. You see the cells which have ARF, they are reduced colony size and colony number. And if they have CARF and ARF together, you have a stronger growth arrest. So then we also looked at Role of calf in senescence or aging. Replicative senescence, as you all know, as the cells are dividing in cell culture dish, they undergo replicative senescence, they stop dividing, and during this phenomena, calf goes up, as is seen here. And if you induce, stress-induced aging in cells, calf also increases. And on the other hand, if you put calf extra copies into the cells, they also undergo senescence, showing that calf has a role in stress and it puts the cells into growth arrest or aging. Now, you would imagine if calf has that sort of function, if we knock down calf or silence calf, or if we take calf away from the cells, would cells divide more? So we did this experiment, as you see here. The calf siRNA put the cells into apoptosis. It did not allow cells to divide more, but induce apoptosis in cells. What does it say? It says it's an essential protein. If you take it away, the cells would die by apoptosis. So this elaborated our experiments further. I'm going to show you here how the calf knockdown cells undergo apoptosis. These are the normal guys here. And if you have the calf knockdown cells here, you would see that they will burst into apoptosis as in the culture dish. This is apoptosis sign. So we had gone further into these to define the mechanism of apoptosis. You see, normally the cells would divide, you know, the chromatids are formed, then the cells are getting into the daughter cells here. But if you have calf siRNA therein, you watch here, there is no division happening here. The cells are staying there as condensed, stage, and there is entering into apoptosis. So this defined that calf is an essential protein, and if you take it away, the cells die because of apoptosis. Now, this was really confusing for a couple of years when we were trying to work out the mechanism of apoptosis, and my postdoc Caroline, she put the expression level of calf into the cells like super expression and overexpression. Then she made the cells with the overexpression. As expected, calf overexpression put the cells into growth arrest. That's here. But she noticed when the cells have super expression of calf, these guys are not entering growth arrest. They are reverting their phenotype. They are entering into cellular transformation. You see increased number of colonies here. So this really confused us that how the over and super expression of the same protein put the contrasting phenotype into the cells. So we went further. You see the overexpression. You see the super expression here. And you see the signaling here is reversed. With overexpression, P53 is going up, and P21, which is a growth arrest inducing protein, is going up. But the super expression is reversed. So this was incredible thing to resolve, and we have gone further to resolve it into the DNA damage pathways, growth arrest pathways, and you see the signalings or markers are reversed between overexpression and stress expression. So let me remind you here, let me recall what I said. Calf is a stress protein. If you induce into the cells are stressed, they're entering growth arrest. But if it is overexpression, it goes beyond the growth arrest. It actually gets cancerous, which tells you too much of stress may lead to cancerous phenotype. The cells would not keep on dying and keep on growth arresting. They would move further. They would get into a more proliferative stage. So we wanted to validate that phenotype, and Raj Kalra, another postdoc and staff member in my team, he looked at the databases and found that most of the malignant cancers, they have super high level of expression of calf. So he went on checking this in the cellular system. He made calf overexpressing and super expressing cells. Here I'm showing the super expression. And if he did that, the cells were increased in their migration. They were becoming more metastatic. So this is all published data, so I'll go quickly over it. So the cells which are now super expressing calf, they have more mesothelial markers. They are enhancing their EMT signaling. And you know how EMT signaling is closely related to metastasis. Now, we took those metastatic cancers in the cells, and then after establishing that super expression is associated with malignant phenotype, we also wanted to validate by knocking it down. And here you see when CAF is silenced here, the signaling is actually reversed. So you can actually reverse the EMT by CAF siRNA, not only at the cell level, but also at the mouse level. Super expression of CAF leads to bigger tumors, and if you knock down siRNA, tumors are regressed to this level. There is absolute no growth. You see the red curves here. There is absolute no growth of the tumors there. And we also did the metastatic, lung metastatic assays where CAF siRNA put it into the adeno-oncolytic virus, completely inhibited metastasis. So just summarizing further, we've also gone further looking for the microRNAs, not only siRNA, and we showed that the microRNAs which target CAF also cause tumor suppressor at the cellular level and at the animal levels too. So what we have shown over the years about this protein playing a role in stress, aging, and cancer is, it is the crucial level of CAF expression. It is a threshold of CAF which determines the proliferative fate of cells, either entering apoptosis or going beyond and getting malignant transformation. So I showed you the link between stress and cancer. Raj went further and he wanted to prove really CAF is induced by stress, and if you do that at different levels by stress itself, your cells might be either dying or they may be transforming into cancerous phenotype. So he took about 16 models of stress, chemical models of stress here listed, and used the cell culture system. This is also published, and what we find out by this experiment is the stress types that do not change CAF level do not change the proliferative phenotype. The stress levels that increase the CAF level put your cells to growth arrest, like this, premature aging. And the stress type which cause super expression of CAF actually make your cells cancerous. So there is a strong message here. If you stress the cells too strong, you are actually making them to cancerous phenotype. He actually looked at the proliferative capacity of cells. Here I'm giving one summary slide that the control cells were like as you expect the control. The stressed cells to the level when the CAF after recovery stays at a bit high level, they are growth arresting, they are entering senescence, and the stress types and the levels, CAF levels, which are staying higher after recovery, they make the cells malignantly transformed. So what I'm trying to say is that the stress has a very close relation with your cancer or carcinogenesis. Now, this is where we meet nutraceuticals. You want to intervene CAF expression levels to the levels that they stay or they are inhibited, and you want to stop them going to super high levels. And one of the examples, as Sunil showed, we've been working on lots of natural resources. Ashwagandha has taken the lead last few years, but we are working in parallel many others. Soya sapiginol is one of them, and we show that it inhibits CAF, and when it does so, you can use it as a tumor suppressor natural molecule. The other one is cucurbitacin B, which is there in bitter cucumber. Goya, we call it in Japanese, and Karela, we call it in Hindi. So cucurbitacin and vidanone. Vidanone comes from ashwagandha. We published that the cucurbitacin B and vidanone combination is super tumor suppressor activity, and it is highly useful for tumor suppression. So like this, Sunil also showed this slide to tell you guys that we work together and we share slides. And so Morlin and CAF have been the subject of our team with the great collaborations with Dr. Sundar, Dr. Ajay, who are here. We've been pouring in bioinformatics, we've been pouring in molecular dynamics, looking at the target molecules and looking at the activities of several natural compounds and nutraceuticals. So intervention by nutraceutical is a great idea, and we know its value already. And I showed you today our work from CAF. It increases during stress. It puts you to premature aging if it is high, and if it is super high, it causes carcinogenesis or tumorigenesis. I think this is my last slides, and I do want to say thank you to the whole team of Lab in Japan and the satellite sister labs in IIT Delhi, IIT Guwahati, Guru Nanak University. Dr. Pati is also here, and this collaboration over a decade, last 1 decade, has done a great work for us and for developing knowledge on these lines. Thank you so much.
Thank you, Reza. So the topic is open for 1 or 2 quick questions. Any questions? Any comments you have? Yes.
Thank you for the presentation. So, what is the level? What is the level which is upper and lower expression?
Great question, great question. So what I've been talking about is the cellular system. We've been trying to define like how many picomoles or nanomoles of CAF would be defined as normal, and how many of these as a overexpression and superexpression, which will link to carcinogenesis. It's an extremely difficult question because different cell types, different cell lines have their own threshold. So it's relative what you're talking about. So when we are saying normal cells, your threshold will be different, and as compared to some other cell types as well. So it's very difficult to say your threshold. We're trying to work on it to see what are the threshold which will be defined as stress inducing. So that's something we are working on. Great question.
Any other comments or questions? If there are, yes, please.
Please repeat. I couldn't get your question. Is it Sadbi? Yes. Okay. Go ahead, please.
So, can you please comment on the expression levels of this protein in different types of cell lines? Mainly, I'm interested in adipocytes, fat cells.
In what cells?
Fat cells.
We haven't looked at fat cells at all. What we have looked at is the different type of cancer cell lines, stressed cell lines, and the normal cell lines. So once again, if you're asking me the absolute levels of CAF, I wouldn't be able to say. You have to take one cell line and see what's normal. You stress it, what's becomes higher, and if you keep stressing it, it becomes super higher. So in other words, I don't know.
So is this protein ubiquitous?
Yes. Somebody has to do it.
Yes, please.
I just want to know whether this particular protein is excreted out or it remains within the cell.
It is not excreted. It goes from nucleus to cytoplasm. That we know for sure. We do not know if it will be excreted if you have extremely malignant cancer cells. There is a possibility.
Yeah, because it may affect the innocent bystanders.
It is. It's clearly a question.
Yeah.
I need next generation to work on it.
Thank you.
Okay, so we'll move on to the next speaker.
Oh, you have a question. Go ahead.
Very nice presentation.
Thank you.
I just want to I didn't hear. So what kind of stress that you are giving?
We use some kinds of chemical models of different stresses: pollution, smoke, metal, temperature. So we use 16 models. I showed one of the slides.
Okay, thank you.
Thank you once again, Renu. And I move on to the next speaker. So I would like to request Professor Agrawal to introduce the second speaker.
Who is the next speaker? Right, Sundar. Yes, I was going to open his.
Hello, friends, ladies and gentlemen. It's my great pleasure to introduce you the next speaker. His name is Dr. Durai Sundar.
Use the cursor.
He is the Institute Chair, Professor and Head in the Department of Biochemical Engineering and Biotechnology at the Indian Institute of Technology, Delhi, here in India. His areas of interest is very diverse. It is in the area of bioinformatics and computational genomics, and design and analyze genomic engineering tools, and develop AI ML based solutions, and that are involved in cancer research, and whole list of them I won't have the time to go through. So with all that, it is my great pleasure to introduce you next speaker, Dr. Dorai Sundar.
Thank you. Thank you very much for the kind introduction. Good morning, and first of all, I'd like to thank the organizers for having given us an opportunity to present our research under the banner of Picnic. And the topic that I've chosen today is drug-membrane interaction studies and how it can lead to new considerations for targeting cancer cells with selectivity. And we all know that, the development of drugs that can be highly selective for cancer cells is very important, which are safe for the normal cells. And one of the key parameters, one of the key metrics that one can look into is by evaluating the ability of the drug to permeate through a membrane. So selective targeting in cancer therapy is important because of the reasons what you can see. Many of these cancer chemotherapy is often associated with severe side effects, and this could be potentially because of the lack of selectivity. And studying the interaction of anti-cancer compounds with specific targets, which are very unique to cancer cells, provides the leads for us to start with. And if there is a way in which you could study the interactions, the pharmacokinetic properties of these compounds in the tumor microenvironment, and that can provide insights for enhancing cancer cell selectivity. And we would like to project here some of our data from how we could get insights into the permeation of drugs, which could be potentially useful for chemotherapeutics. So when you look into the selective targeting of cancer cells, right, one way of looking at this is when you look into the pharmacodynamics part of it, where we look into molecular targeting, which would be straight-jacketed work where you look into the target proteins that are unique to cancer cells. We also look at target proteins that are overexpressed in cancer cells, and here heterogeneity is a challenge. So the drugs that can target multiple targets have been reported to be quite promising. But I would like to draw your attention to the pharmacokinetic part of selective targeting of cancer cells, where we are looking into, I think I'm not able to see it clearly. Let me see the slides here. So targeting the small structural or physiological differences existing between cancer cells and normal cells. Is there a way we could learn from that, which could be important for targeted delivery? And there are also reports on how a cancer cell membrane looks like and how that changes the dimension in which a potential drug could selectively permeate through a cancer cell. So what I'm going to present maybe in the next 5 to 10 minutes is the drug membrane interactions that were studied and how we could identify some kind of a selectivity in the chosen drugs. So I'm not going into the detail of this particular slide. So there are quite a lot of interactions that actually can happen when a drug is actually in the membrane. And right, I think these are textbook information. But there are a lot of things that one can learn from how a drug interacts as it goes through the membrane. And important information related to absorption, distribution, metabolism, and so on. All these information can be easily derived today by performing computational experiments to derive some insights. And this is the barrier which actually needs to be passed by any drug molecule. And one can easily achieve many things, which is very, very important to understand the permeability. So when you look into the membrane, and I think I'm not again going into the details, one of the important things that we need to understand is the asymmetric distribution of phospholipids. So the lipid bilayer which is there in the membrane and the composition of it. So there are quite a few that have been reported. Right, we have this phosphatidyl choline, then sphingomyelin, and so on. So there are multiple such composition which is reported for membranes, and I think there are also varied distribution of these. And when you look into a normal cell and a cancer cell, there are quite variations that have been reported. So one of the things that we could do to understand how a drug permeates through a membrane is to understand the free energy profile. So if a drug moves through the membrane, the kind of interaction that happens, so on a scale, you can actually look at the interaction of a drug, let's say from outside the membrane, through the membrane and inside the cell. So there are ways in which these can be measured, a drug moving through the membrane, and we can calculate what is called as a potential mean force. So that basically tells us if you want to measure the permeation as a function of a distance or as a function of the different composition of the lipids which is going to be making these membranes. So today with modern computational techniques, one can do what is called as an umbrella sampling. So there are ways in which you can place a drug on, so you can build a membrane with whatever composition you want based on a particular composition. So here in this study, what we are reporting is a membrane which is largely composing of phosphatidylserine as one of its major components, and that has been reported in several cancer cells. So you can do what is called as steered molecular dynamics. So without going into the details, so to put it in simple terms, so if you are going to have an extracellular side of a membrane, you can build a membrane with whatever composition you have. You can have an outer leaf, you can have an inner leaf, and then you can look at a drug passing through the membrane, and you can see what is actually happening to its trajectory from outside to inside. So you can actually have a force, you can actually push it from one side and pull it from the other side, and you can look at the trajectory, and this is what we look at the calculation of potential mean force. And there are some standard measures where you could calculate the permeability coefficient and so on. And this actually, as I said earlier, comes from the kind of asymmetry that is lost in cancer cells. So when you look into normal cells, the distribution of the phospholipids on the membrane, it's pretty asymmetric. But this becomes quite symmetric in cancer cells, that is what is reported. And this is one of the data that we have used for preparing or building a membrane. And that is what I'm actually trying to summarize here. So there are ways in which, I think, this is a standard pipeline where you look at identification, hit identification, lead optimization. So this is where this kind of a study can intervene and throw some insights into whether the drug or the potential molecule that you have identified can really permeate through. Because there are a lot of papers which talks about how they interact, the potential drug target interaction and so on. But many such things fail. An evaluation of this parameter, whether it can pass through a normal cell or it can be very selective for a cancer cell. So this kind of a study which can provide insights into preparing cells with different compositions of phospholipids can be very, very helpful. So what we have done in this study that I'm reporting is, so we have made a composition of a lipid membrane for a particular cell membrane that looks like a cancer cell, which can potentially modulate the drug activity. And we have looked at ways, so this is one of the very first such reports where we have tried to build a membrane with different compositions. I'm not going into the details of the different membrane compositions, so I'm only presenting the data from one of the phospholipids, which is phosphatidylserine, and the details are here. So the phosphatidylserine is usually present in the inner leaflet of the membrane, so you can dissect the membrane into different sections, and you can create composition of these membranes based on what could potentially look like a cancer cell membrane. And that is how we did in terms of the number of molecules that were placed on the inner leaflet and then the outer leaflet and so on. And then we exposed this lipid bilayer membrane to a particular drug. We pushed it from outside and looked at its trajectory through the membrane and see whether it is able to enter into the cell. So, so we built this. So this is just a summary of what we have built. So we have built a normal and a cancer cell membrane. based only on phosphatidylserine exposure. So there can be many different ways to make this. This is one of the pilot studies that we have started with, and we looked at, right, I think, the composition, and we also throw in some of the other key ingredients for a membrane. But the major thing that we are looking at is whether it can pass through this particular membrane. So as Dr. Vadra mentioned earlier, we have been studying on some of the natural compounds. So the ones that were chosen for this study included the ones from ashwagandha, which is vitaferin A and vithanol. And we also looked at two other compounds, which is one is called as the CAPE, the caffeic acid phenylethyl ester, and the atropine C. And I think the slide is not clear here, but basically we had some data in terms of some selectivity, which was actually obtained from experiments, where some of the natural compounds were highly selective for cancer cells, and some of them were actually safe for the normal cells. So with this data, what we started was we built that membrane, and I think what I'm showing you here is the differential permeation, which has been calculated by the measurement of what is called as a potential mean force. So you're seeing these four panels: vitaferin A, vithanol, CAPE, and atropine C. And one on the blue color, light blue, the cyan, is the normal cells, the trajectory of the drug through the membrane, and what you see on the red is the cancer cell, the membrane through which the thing has passed. And the first inference, I'd like to draw your attention to this peak here, which actually is very clearly showing the hydrophobic core of the membrane. So you can see, right? I think this is the x-axis shows you the length of the membrane, and what you can see here is the hydrophobic core of the membrane, which is seem to be creating a major hindrance. So this is for vitaferin A, vithanol, and similarly the same is the case for CAPE. So there seems to be some kind of an hindrance there. And then when you look into what's actually happening to the atropine C, it's kind of the head group, the lipid head group seems to be the major hindrance. So the principal hindrance for different molecules are different. So that means this will also decide the way the drug is going to move through the membrane. And similarly, we went on to measure some of the key parameters that are the free energy change and so on, which are generally used to measure how a drug is able to move into the membrane or it is not able to move into the membrane. And right, I think let me go into the other way, right? I think one, actually we looked into the diffusivity, which is the direct measurement of permeability. We also looked into the resistivity, in terms of how a particular membrane with a certain composition is able to resist the movement of the drug through the membrane. And that was also quite corroborating with what we got in terms of the permeation. So the summary is, if you have a membrane which looks like a cancer cell membrane, in this case, it is largely made with one type of lipid, which is phosphatidylserine. It caused some change. So it's a very preliminary study. It changed the energy landscape, whether it is right in the middle or through the end of the membrane towards the inside. So the models that we have developed and we compared from the normal and the cancer cell, that clearly showed that there seems to be some kind of a selectivity of a certain type of natural compounds. In this case, it happens to be vitaferin A, vithanol, and CAPE, where there was a differential permeability in terms of the normal and cancer cell. And this selectivity can be explained with a difference in the free energy landscapes at the cell-water interface. So because solvent is also going to be the one of the key players. So you could measure this as a function of distance or the different compositions of the membrane or even the environment where it is going to be. So this, though may not be a very complete study, but it really brings in some kind of an insight. So we started off with some experimental data that we had in terms of selectivity, and we were able to add more value in terms of how there seems to be a difference in terms of selectivity towards the cancer cells, which is very, very helpful. So to conclude, I would like to say that, I think one can study different such membranes, build different such membranes to understand how drugs are able to move. And this difference can be very, very helpful in terms of optimizing the drug, right? In terms of, because we also, I'm not going into the details because the different compositions, each drug is going to have a particular structure. The different parts of the drug molecule interacts differently to the different composition of the membrane. So all such details can be easily be obtained, which can straight away tell you about the selectivity. So one can be even planning their experiments before getting into the nitty-gritties of whether it is going to work or not. So in terms of future directions, where we are heading towards this, we are trying to now build. So we have actually have many such models where we are trying to make with different lipids which have been reported in literature. So we are looking at, and I think one thing which we could see is there is not much literature on the composition of such cancer cell membranes. So that would be very, very helpful. So these are being built in the light of these preliminary studies. So this has been recently published in journal Membranes, and different class of drug-like compounds can be studied so that these can be used as a very, very important consideration before the drug design and further processes take place. Thank you very much, and I'll be happy to take any questions.
Questions? Yes.
Yeah, like you have so many proteins that interact with the lipid membranes. And in cancer cells, you know that efflux proteins are very highly expressed in the drug. It's not a normal mechanism of movement of drugs. So how do you plan to address all these?
So I think this, as I said, was only concentrating on lipid composition. There are also other reports that we have done where we have looked into all possible potential interacting factors. So that could be easily added to the membrane. So it could be done. So we can pretty much mimic a real membrane looks like. So the objective of this study was only looking into the lipid composition. That broadly tells you whether it is going to move or not. But then I think other factors will come in terms of its potential passage through the, yes. Because if you see, some of these things could not even move through a normal cell membrane. So that actually is a very good thing to start with. So very valid point what you said.
Hari has a question. Hari, here.
It's a wonderful presentation, and what a nice concept to look at differences in the membrane, lipids, especially phosphatidylserine, which is in at least which is at least overexpressed in 7 different phenotypes you showed. And I think if you can add, as was just presented, other proteins and glycoproteins which form the complex on the membrane surfaces, that will make this model really more applicable.
Sure.
But beyond that, if you look at solid tumors, there's a high pressure, intratumoral pressures, which is one of the limiting factors for our drugs to go there. So if we can model that and how we can use that information to have drugs more targeted, wonderful model. Enjoyed.
Thank you very much for your inputs.
Yes.
Thank
Can I ask?
Yes, please.
It was a wonderful talk. My question is that different drugs at different concentration might be acting differently over the cell membrane. So starting off with such an experiment, how do you actually decide that this is the particular threshold, or have you considered this thing a different concentration and this whole change that about?
So I think, so in fact, we have published a paper last year where we have, I think it's a valid question. So this has been one of the things that has been in our mind. So we have always been debating with our experimental collaborators about concentration. So, I think concentration makes a difference in experiments. So how do you do that here? So we have looked into, so when we have thrown in different populations of these drug molecules and we can track it. So eventually what we have seen is, it is taking a similar path. In terms of path, right? We have even looked into keeping them at different parts of the membrane and then trying to pull down. That has also not made a difference in terms of the tracking.
Yes.
So the objective, as I said, here is only to see whether it is even able to move.
Move or not.
So I think that is a very valid point, and it is there in our mind for quite a long time.
Yes.
Yeah.
Thank you, sir.
Thank you.
One more question.
Thank you for the presentation. Cholesterol is known to increase membrane flow
Yes.
in normal cells also. So how do you think cholesterol would make your results a little
So
different?
we have added, so I'm not going into the details, actually it is. So we have pretty much tried to add as much, so in terms of number of cholesterols, we have added all of this. So, so pretty much it is there in the study. So only thing is we have kind of played with how many molecules will be placed where, whether the outer leaflet, inner leaflet. So that is something we have done quite a few background study on this. Thank you.
Thank you, Dr. Sundar, for such a Sundar talk. We all enjoyed it.
Thank you.
So we have next speaker in the series, Dr. Fayaz Malik. He is currently professor at CSR Indian Institute of Integrative Medicine in Srinagar. His major research interest is to understand the fundamental regulatory mechanisms associated with tumor metastasis and therapeutic resistance in TNBCs. He started his career, his major awards are in 2009, he got the National Young Scientist Award. Then in 2014, he got the National Swarn Jayanti Prestigious Fellowship Award from DST. And then in 2015, he had the National Bioscience Career Development Award. Today, he is going to talk about the TNBC, and the title of his talk is Discovery and Preclinical Development of Small Molecule Targeting Paclitaxel Resistant Cancer Stem Cells in TNBCs. So over to you, Dr. Malik.
Thank you for a nice introduction. So I thank the organizing committee and their lab for giving me the opportunity to be here to present my work. It has been 3 years I have not traveled out from my house. So, and thank you to all of you for giving me this, forceful opportunity to be out of my home and take a travel till Delhi. So I'm not able to find presentation aids.
Talk is on the desktop. Just open your phone. Open your phone. Desktop. Your name is there.
All right.
Yes.
Yeah. So today I'll be talking about how I have been working on particularly the growing concern, the highest incident cancer is the breast cancer. The major focus in the breast cancer remains the triple negative breast cancers. So my talk will be more about how we try to understand the fundamental biology related to its aggressiveness and how we are actually looking for the natural product small molecules to actually target this cancer. So I will not, because this is a very common slide, everybody knows, but because students are around, so I just want to make them aware that the breast cancer has been it is the largest incidence in the world. It is number one cancer in terms of the incidence. So with, and the mortality, and not only globally, but coming to India. So from 2 years back, it was a cervical cancer which was on the highest incidence in India, but breast cancer is now overtaking the cervical cancer and is number one cancer in India. So the growing incidence of breast cancer in India is again a concern which we try to look and address locally. And before coming to the incidence, and I will just try to brief you about, like, the breast cancer has clinically been actually divided into 4 different subtypes. It depends upon the expression of the hormones, ARPR. So this is luminal A, luminal B, which are like, which have ARPR receptors present on them. So there are aromatase inhibitors which are have been very successful, and they are not more aggressive cancers. And there is a HER2, which the epidermal growth factor is there. So Herceptin is the blockbuster drug, and other drugs have been there for this subtype of cancer. And third one is the triple negative breast cancer, and which doesn't have all these receptors. It is ARPR negative and HER2 negative. It is the most aggressive cancer subtype. So the only chemotherapy and radiotherapy is the only option for those patients, despite certain agents have been tried for target-based therapy, but have not been successful so far. So why I'm most interested in working for triple negative breast cancer is because compared to United States, India has the highest incidence of this type of subtype of cancer. So TNBC is the highest in India. So compared to 12 to 15% incidence in the US, India has 28% incidence of triple negative breast cancers. So that's an alarming situation in India. There's a lot of literature has been shown it now that triple negative breast cancer burden has been increasing in India, particularly in the young ladies. The concern is the ladies which are below the age of 40 are actually suffering from this type of cancer. There's a lot of literature has been recently published how this cancer is actually making this space in Indian women. So why triple negative breast cancer is more aggressive? So I just want to add on a few things here, because this TNBC is more has a more non-undifferentiated type of cancer. So you can see that, as I said, there's a different type of cancer. There's a Claudin low, there's a basal, there's a HER2 and luminal. So luminal cancers are more differentiated cancers. HER2s are also kind of differentiated cancer, but this triple negative breast cancers are their undifferentiated cancers, and they are more similarity towards the when the memory during the memory development, when the cells start becoming from stem cells to progenitor cells to myothelial cells and differentiated cells. So based on the similarity between the normal breast development and the cancer cells, you can see the triple negative breast cancers fall on the top, which are more closely towards their characteristics are more similar to their stem-like cells. So what are these stem-like cells? They are 0.01 to 1% of cells existing in all the tumors, and they have been shown from literature they have powerful selective power. They are highly immune motile. They are responsible for rectness, and the cells have a poor clinical outcome. So studies have shown that these cells, a lot of literature has come out from last 2 decades that these cells are the origin of metastasis and non-responsiveness to the chemotherapies. So in particularly TNBCs, so 70 to 80% of the TNBCs are actually, they have P53 mutations, and 40 to 50% of the TNBCs are P10 mutations. And the patients who have the double mutation of P10 and P53 is the most aggressive subtype of this TNBC. So it has been shown that when there's a loss of these tumor suppressors, so the level of AKT is very high because it falls down, because P10, these are more P10 is a tumor suppressor, it doesn't phosphorylate PI3K to phosphorylate AKT. So when this P10 is mutated or lost, so its AKT expression goes up. So it has been shown that the higher AKT expression leads to poor prognosis of this TNBCs, whereas low AKT shows a better prognosis. So it depends upon patients. So the question in our mind came, so this AKT has actually 3 isoforms. So we try to understand what nature needs to make the 3 isoforms of this AKT. So we try to understand how each isoform of AKT plays a role in aggressiveness and TNBCs and how this is affecting the stem cells. So it was our collaborative work, our British counterparts. So we took breast cancer samples from British origin samples, and we also took breast cancer primary samples from the Indian origin, and we tried to do immunohistochemistry and see what is the expression of 3 AKTs. And we found it's a primary tissue, primary breast cancer tissues, the AKT1 level was quite high, and it was found both in the British samples as well as in Indian samples. So, while other expression of other AKT2 and 3 was not as high as there was AKT1. So, but when we took the metastatic samples, so it was some British liver, normal tissue, it's a liver metastatic tissue, took a lesion from the liver metastatic site, and look for again the expression of 3 AKTs. We found AKT2 was high in the metastatic sample compared when it was compared to this normal tissue in primary breast tissue, primary breast cancer, AKT1 was higher, where in metastatic sample AKT2 was high. So similarly, we also found, we couldn't find liver samples in India, but we found some non-metastatic samples of breast, and we found here it was, the samples were taken from the Gujarat Cancer Institution. And we again found in non-metastatic samples, AKT2 was quite high. So then we try to understand what different AKTs actually, different isoforms of AKT play a role, and how, how they behave when we put in the cell. So we took MCF10A because MCF10 is like, immortalized normal cell, and this BT549 is like a triple negative breast cancer cell. So we try to understand by inhibiting the expression of all three AKTs and see what will happen. So we did this individual knockdown of AKT. We also did a double knockdown of AKTs, but I'm showing the data with double knockdown. So when you did a knockdown of AKT3 and AKT1, so only AKT2 is present here. So you can see the invasion and migration of the cells actually increase when the AKT2 was there. So again, in this cell line, when we knock down AKT1 and 3 and only AKT2 is there present, so you can see the invasion and migration potential of these cells was considerably increased compared to other cells. Okay. Sorry. So now I try to understand, there's a defined stem cell markers, and there's a, the established, protocol for analyzing the stemness of those cells, like those membranosphere assay. So these cells, when they grow in the single cell, they form spheres. So we did a sphere formation assay, membranosphere formation assay. We perform it with these cells. So there are cells where there's a double knockdown of AKT1 and AKT3, means when only AKT2 is present. So you can see higher number of membranospheres were formed there. And similarly, there's another assay, is ALD8, is a stem cell marker assay. So normally it shows is a marker of cancer stem cells in TNBCs, other cells. So when we knock down individual AKTs and we knock down both the AKTs, so the point here is when there was AKT1 and 3 was knocked down and only AKT2 was present, we can see the population of stem cell was expanded, considerably from 4% to 12%. So showing that the role of presence of AKT2 only actually enhance the stemness in those cells. So it was very interesting to first, we try to understand, there are many experiments where we keep on proving how these individual AKT isoforms actually affect the stemness in breast cancers. And then we screen many chemotherapeutic agents and see how these individual AKT isoforms actually respond to this. So it was like quite amazing to see here when we knocked down AKT1. So cisplatin was unable to induce apoptosis in those cells. So you can see here the caspase, cleavage caspase. When there is knockdown of AKT2 and 3, the caspase is cleaved, and this PARP1 is like an apoptotic marker, the cleavage of PARP. So when there was no AKT1 isoform, the cells couldn't go through the apoptosis and cells were alive, while other cells could die through the cisplatin. And then what we did, we enforced it. We put that copy, we put the AKT1 overexpression in those cells by putting lentivirus here, which is expressing AKT1, and we could see by putting AKT1, overexpressing AKT1 in those cells, these cells could actually respond to the cisplatin and can undergo to the apoptosis. You can see that the cleavage of caspase and the cleavage of PARP. So showing that, this slide actually shows that the expression of AKT1 could be actually a biomarker to understand whether the treatment of cisplatin can be effective in the patients who have loss of AKT1 or who are mutated AKT1, where there's AKT1 is not functional. So this can actually indicate that analyzing the expression of AKT1 in clinical samples would be very informative to design the treatment. And then we put the same experiment in the mice and see when there's a knockdown of AKT1, the cells could not respond to cisplatin, the tumor growth is set, tumors are growing well. Well, knockdown of other AKT2s, the tumor was actually reduced. So again, showing that the knockdown of AKT1, when there is no expression of AKT1 isoform, this mice were not responding to the cisplatin treatment. So then we try to understand what is the reason that this AKT1 is actually making these cells non-responsive. So we did a number of experiments and ultimately find that there's a ABCG2 protein which is actually a flexing FlexCom protein. So we found that the ABCG2 was under the knockdown of AKT1, this ABCG2 expression was going high and it was actually flexing the drug out of the cells. So when we put the cisplatin plus knockdown by siRNA, by knocking down ABCG2 protein, we could actually restore, the apoptosis in those cells by putting the cisplatin in cell. And the same was done in other cells also. So since the idea was actually to look for the individual AKT isoforms. So we tried to establish an assay in our lab, where a kinase assay, where we actually screened around 700 molecules from our national product repository, and we found like there are 2 hits which can actually selectively target AKT2, and they are not targeting. There's like a good fold change between them, like AKT2 it's only 1.48 nanomolar, while AKT1 is 90.1 nanomolar inhibitory potential. So we took this molecule for further studies and tried to understand how it is targeting those cancer stem cells. So the red ones are here cancer stem cells, and this that's what we are actually looking to target them to have a long-lasting response. So in this case, what we did is we did the experiment. This experiment has also been done in the literature. So we treated the triple-negative breast cancer cells with the paclitaxel. So, and this is the stem cell population. You can see here in the control, there is 6.2% cells are stem-like cells. But when we put the paclitaxel, their population goes up to 20%. So it means the relative population was increased because the paclitaxel was only targeting the differentiated cells, where this population of cells was not getting targeted. That's why the relative population of the cells got increased from 6% to 21%. So, but when we took the molecule which we identified in the previous study and put in those cells, you can see we put the paclitaxel plus the molecule. So we can see the population came down from 21% to 6%, to 2%, 81%, 0.2% at a different concentration of this natural product molecule. So at 50 micromolar, the population came down from 22% to 0.76%. So it was actually effectively killing those cancer stem cells which were not able to be killed by paclitaxel alone. So this is another assay which shows the self-renewal capabilities as a mammocepher assay. So we again see that paclitaxel was not able to take care of those cells, while IM152 plus paclitaxel was able to kill all those cells. So these are the stem cell markers. You can see the OCT4 and SOX. By the treatment of this molecule, the expression of these stem cell markers was considerably went down in both the cells, SUM159 and MDMB231. So then we put a mice experiment again, and we try to see whether this molecule was able to inhibit tumor growth. It was able to inhibit the tumor growth, slow the tumor progression at 50 mg per kg concentration. Not only that, we excise these tumors and subject them to flow cytometry and try to understand what happened to stem-like cell populations. And we see that IM152 treatment in the mice brought down stem cells from 13% to 6%. So again, showing that our in vitro data was verified that this molecule is actually targeting stem-like cells in these breast cancers. So we found this molecule very safe. So we did a pathology of all the vital organs. This molecule was not causing any damage to cells. And we found a very promising pharmacokinetics profile of this molecule. And This molecule we have currently submitted patent, and the CSR is the organization which they have selected this molecule for the IND filing. So we are undergoing preclinical studies for this molecule right now. And this is the team who was working with me. Thank you.
Thank you, Professor Malik, for the nice presentation. You can ask some questions. Yeah.
Yeah, it is a nice presentation. My question is about stem cell marker in MDMB231 cells. How did you measure? Did you isolate stem cell population from MDMB231 cells?
Yeah, there are set protocols for that. So there are different markers. There is CD4424.
So what the population do you get out of those MDMB231 cells?
Yeah, normally if we put ALDH as a marker, if you use ALDH, it's only like 4 to 6% population are only stem-like cells. So that's 96 are the more differentiated like cells. Yeah.
Another thing I noticed that you did some work in BT549 cells.
Yeah.
Those were cell line work.
Like,
But for the mice, you selected 41.
Yeah.
What was the reason you selected two different cell lines?
The reason is because BT549 is a human's TLBC, but 41 is a mouse BTC. But the reason why we put 41 is because I don't want to use immune-compromised mice. I want to use immune-competent mice to replicate what is actually the condition in human beings. So that's why we put 41 a human mice cell line in a immune-competent mice biopsy. So to be a relevant model here. Yeah.
And about one basic question about the small molecules. How do you decide that your molecule that you identified is a small molecule? It is not a.
Our institute has it has almost 1 million small molecule repository. There's a huge library there. So we keep on screening, and we have assays, certain kinase assays where we keep on screening. So I set up this kinase assay of AKT3, 1, 2, 3, and then screen all molecules. The first we go through the virtual. So we do some computational biology where we identify these can be probable hits, and then we do. Yeah. Yeah. Is that all? Thank you. Yeah.
Thank you. Presentation. So we move on to the next speaker. Our next speaker is Dr. Ajay Kumakara, and he is a professor at the Indian Institute of Technology, Guwahati. He had his PhD in biochemistry from Amala Cancer Research Institute, and then joined Guwahati as an assistant professor in 2012 and as an associate professor in 2015. He has developed novel drug targets for oral cancer, and he developed different formulations of drugs for better drug delivery and bioavailability. And his area of research interest is role of inflammatory pathway in cancer development. So, Dr. Ajay, it's all yours.
It's on the desktop.
Yeah, but it's not moving. It's cursor. Sounds showing internet connections. No, I think someone's hanged or something. Okay. So, very good morning to all of you. First of all, I would like to congratulate the organizing team, especially
Professor Alok Sibardi and Professor Anju Srivastava for organizing this wonderful event. So today I'll be talking about new natural solutions for treatment of oral diseases. And oral diseases are the diseases of the oral cavity, and 90% of 90 to 95% of the population experience one or more type of oral diseases in their lifetime. There are different types of oral diseases such as oral submucosal fibrosis, gingivitis, oral mucositis, oral erythroplakia, oral leukoplakia, oral candidiasis, oral lichen planus, periodontitis, dental caries, and oral cancer. So one of the most of these are inflammation related diseases, and one of the most important or serious oral disease is oral submucosal fibrosis. So what is oral submucosal fibrosis? It is a chronic, insidious, and pre-malignant disease of the oral cavity, and it is characterized by the deposition of dense collagen in the connective tissue. And this condition actually was described by Susruda, and he coined the term as Vedari in Indian ancient medicine. And this is a potentially malignant disease and was first reported in the people of Asian descent in the early 1950s. So in India, the prevalence rate is almost 0.5%. That means 50 to 70 lakhs of our population are affected with this disease. So, but the most dangerous part is that many people of oral submucosal fibrosis, they can develop to oral cancer. So in India, almost 7% of the oral submucosal fibrosis patients develop into oral cancer. And this is a multi-stage process. When you chew tobacco or arik nut, first some staining happens in the teeth or oral cavity, then slowly inflammation happens. Finally, then fibrosis happens. Then you will not be able to open your mouth as usual, and then slowly it develops into oral cancer. So therefore, suppression of, because OSMF leads to oral cancer. So if you suppress OSMF, you can, it prevents oral cancer. So what are the treatments available for this disease? Actually, there is no effective treatments till today available for this, inflammatory condition. And the most common treatment is the use of steroids, but this use of steroids are not avoid of serious adverse side effects. So what are the risk factors of this disease? Actually, arik nut chewing and as well as tobacco chewing are the main risk factors of this disease. Arik nut contains a chemical called auricolin, and it can induce fibroblast proliferation, and then collagen synthesis finally leads to oral submucosal fibrosis. So how does this risk factors induces oral submucosal fibrosis? It is well established that through inflammation, these risk factors like auricolin or arik nut or tobacco induces OSMF. So when you chew tobacco or betel quid, your epithelial cells get damaged, and then lot of production of cytokines, pro-inflammatory cytokines, IL-6, IL-8, TNF-alpha, etc. can happen, and this leads to oral submucosal fibrosis. So this is a paper published in 2015. They show the serum cytokines levels like TNF-alpha, IL-6, and IL-8 in oral submucosal fibrosis patients. And you can see that the levels of these pro-inflammatory cytokines are very, very high in oral submucosal fibrosis patients. And this is another study published. The level of TNF-alpha in oral submucosal fibrosis, you can see that in controls, it is around 6.93 picogram per ml. However, the patients with oral submucosal fibrosis is 23.46. It is fourfold higher than the normal level. So all of you know that TNF-alpha is the most potent activator of the transcription factor NF-kappa B, and the NF-kappa B also related with inflammation and other chronic diseases. So there are a lot of studies published on the expression of NF-kappa B in oral submucosal fibrosis. This is one paper which shows that you can see on the left-hand side, this is a normal mucosa. The NF-kappa B staining is very less. However, on the right-hand side, you can see the tissues from oral submucosal fibrosis where the NF-kappa B level is very, very high. So that means safe, efficacious and affordable TNF alpha and NF kappa B inhibitors have high potential in the prevention and treatment of oral submucosal fibrosis. So which is the most potent inhibitor of TNF alpha and NF kappa B? Almost 25 years ago, it is published that curcumin is one of the most potent inhibitor of tumor necrosis factor alpha and NF kappa B. This paper published in 1995, and now you can see if you search the word curcumin and TNF alpha, you will get more than 1,000 publications in PubMed. Same goes to NF kappa B. That shows the effectiveness of curcumin in inhibiting TNF alpha and NF kappa B. So all of you know what is curcumin. It is isolated from the plant turmeric. These are curcumin roots, and I'm sorry, there are some issues with the slides. Then the curcumin turmeric roots, and you can see the yellow color, and this is the yellow color that is present in the turmeric roots is the curcumin, and this is the chemical structure of curcumin. And it is not a single targeted agent, it's a multi-targeted agent. It can modulate different inflammatory cytokines, growth factors, transcription factors, apoptotic regulators, etc. And this is one of the paper we published in British Journal of Pharmacology in 2017. So what about the efficacy of curcumin for the prevention and treatment of oral submucosal fibrosis? There are so many studies, we and other labs published, preclinical studies published from other labs also shows the efficacy of curcumin in the prevention and treatment of oral submucosal fibrosis. There are so many studies, more than 100 studies, preclinical studies, and some of them are listed here. For example, gingivitis. And these are the studies published, and you can see most of these studies shows that this compound inhibits inflammatory pathways and inflammatory cytokines. And there are so many studies published on oral candidiasis, oral erythroplakia, oral mucositis, and periodontitis, preclinical studies on curcumin, and almost all studies shows the effectiveness of this compound in the prevention and treatment of these diseases. So what about oral cancer? Yes, there are so many in vitro and in vivo studies on curcumin for the prevention and treatment of oral cancer. Some of them are listed here, and where many studies used curcumin alone, and many studies used curcumin plus other chemotherapeutic agents like cetuximab, geftinib, then some of the natural agents like green tea, then chemotherapeutic agents like paclitaxel. So many studies shows that curcumin in combination with these agents sensitize oral cancer cells to chemotherapeutic agents. So what is the mechanism of action of curcumin oral diseases? So curcumin is known to modulate different pathways in oral cancer, oral diseases as well as in oral cancer. These include different apoptotic pathways, ERK signaling, JNK signaling, JAK-STAT signaling, NF-kappa B signaling, AKT signaling, VEGF signaling, TGF-beta signaling, Wnt signaling, etc. So far I have talked about the preclinical studies of curcumin. This formulation shows an improvement of 70% in the treatment of oral submucosal fibrosis. The next study shows the efficacy of curcumin in the treatment of oral submucosal fibrosis. This was a randomized clinical trial. They have used 2-gram dosages of curcumin and for 3 months, and the age of participants were 18 to 50 years. And then the test group shows increase in mouth opening compared to the control group. Not only that, there was a decrease in burning sensation on visual analog scale with a spicy and normal food. And this study shows that more than 85% of improvement in oral submucosal fibrosis symptoms. Then another study, recently shows that the efficacy of curcumin for oral submucosal fibrosis, and here 80 patients were enrolled in this study. The duration was 3 months, and this study shows that there was a significant clinical improvement in mouth opening and subjective symptoms like burning sensation, pain associated with the lesion, and tongue protrusion in the curcumin group as compared to the control group. And there are many other studies also showed the efficacy of curcumin. Again, this published in 2021 for the prevention and treatment of oral submucosal fibrosis. Here they have used only 50 milligram of curcumin, and this study demonstrates the efficacy of curcumin and topical steroid to alleviate the clinical findings of OSMF. However, the most important part is that curcumin is comparatively more efficient than steroids for the treatment of oral submucosal fibrosis. And this another study recently published of the efficacy of curcumin on oral submucosal fibrosis. All of them are clinical studies. So not only for oral submucosal fibrosis, there are a lot of clinical studies published on the efficacy of curcumin for other oral diseases like aphthous stomatitis, gingivitis, oral erythroplakia, oral leukoplakia, oral mucositis, etc. And almost 99% of the studies shows the effectiveness of this compound for the prevention and treatment of this disease. Again, oral lichen planus, periodontitis, etc., etc., etc. So most of these oral diseases, curcumin shows very, very effective. And last year we put together all this, whatever we published and others published on oral disease, on the effect of curcumin or oral diseases, and we published this in ACS, Pharmacology and Translation Science. The title of the article was From Simple Mouth Cavities to Complex Oral Mucosal Disorders, Curcuminoids as a Promising Therapeutic Approach. So not only curcumin, we have we are working on so many other natural products in our laboratory at IIT Guwahati for the prevention and treatment of oral disease and oral cancer. These are some of the agents. Due to the paucity of time, I will not be able to explain everything, but a couple of recent work I will just show. So, there is a compound called butene. It is isolated from the plant toxicodendron from vernicillin. So we have shown recently that this compound is a very good scavenger, and the antioxidant status is almost like curcumin only. And this compound inhibits the viability of oral cancer cells, and it inhibits the survival of oral cancer cells. Then it induces the apoptosis in oral cancer cells, and it inhibits the migration potential of oral cancer cells. Then it inhibits the invasive potential of oral cancer cells. And interestingly, we have also shown that this compound inhibits NF-kappa B and NF-kappa B regulated gene expression in oral cancer cells. So, in the first talk, Professor Renuvadua talked about mortalin, role of mortalin in oral different diseases. So we have also analyzed the level of mortalin in oral cancer tissues, and we found that mortalin is highly overexpressed in oral cancer tissues. This B graph shows the expression of mortalin in oral cancer tissues. And then C graph shows the expression of mortalin mRNA in oral cancer tissues of northeast region of India. So both these studies show that mortalin is highly overexpressed in oral cancer. And we have knocked down mortalin. And when we knocked down mortalin, we found that it inhibits the proliferation and survival of oral cancer cells. And that also inhibits the inactivation of mortalin, suppress AKT-mTOR pathway, which is highly overexpressed in oral cancer. So later we have shown that Vidania somnifera, vidaferin isolated from Vidania somnifera, also inhibits mortalin, as mentioned by Professor Renuvadua in the previous talk. So we have treated the oral cancer cells with Vidafrin and found that this, Vidafrin shows high efficacy in the in inhibiting or suppressing the proliferation of oral cancer cells as well as inducing apoptosis. Later, we showed that Vidafrin A inhibits mortalin in oral cancer cells and inhibits AKT-mTOR pathway. So in conclusion, inflammation plays an important role in the development of oral disease and oral cancer, and TNF alpha and TNF kappa B and other inflammatory pathways are highly overexpressed in oral disease and oral cancer. So natural TNF alpha and NF kappa B blockers have high potential in the prevention and treatment of oral disease and oral cancer. So clinical trials of curcumin, many clinical trials are already done, but other compounds, more preclinical and clinical trials are required. So this is my acknowledgement. I acknowledge the Dyer Center as well as the DBT, DST, and ICMR for providing the financial support. Thank you. Thank you so much.
Thank you, Ajay. So the topic is open for a quick 2 questions, if anyone. Yes.
Ajay, it is a nice presentation.
Thank you.
My question is about, did you check this mortalin expression in clinical samples?
Yes. So we have used clinical samples from northeastern region. So hospitals in northeastern region, we collected the samples and then isolated the RNA, and we have, that's what I was showing in the C graph.
And that was, those samples were specific to the northeast region, not in other parts of India?
No, northeast region. Yeah.
Yes, Dr. Ajay, very quick question.
Yeah.
We see a black turmeric also in the northeast region. Have you explored its potential?
Not yet. We will do it soon.
So with this, I would like to thank Ajay for a nice presentation, and we move on to the next speaker.
Sunil, can I ask a question? Questions.
Sorry.
It's a very nice presentation, very comprehensive and very viable approach to address a very clinical relevant problem. So I have 2 questions, short questions. The beginning you talked about pro-inflammatory cytokines in blood. So did you check in saliva and is it?
Mainly these are in blood.
Okay. So then second question is that normally look at the biology of the disease like this. There's an inflammatory stage. That is where you probably steroids will work better. But once it is moved to fibrotic stage, it's more like a irreversible type of thing. So are you testing this in the curative intent or in therapeutic intent?
So we want to explore both, because the first idea is that many people are using Incd2022 as well as Waco, and they can develop oral cavity or cervical fibrosis. So before reaching to that stage, we are and patients who are already cervical fibrosis, we want to prevent and suppress that. Otherwise, many patients will develop oral.
Thank you.
Regarding your query to salivary biomarkers, the same parameters which Dr. Ajay has studied, we have collaboration with that lab, and we have studied same nutraceutic, same natural products in the cell line, and we have analyzed salivary levels of those biomarkers. So they could go hand in hand. Yeah, exactly.
Any other questions? Yes. One quick question. Wait.
Sir, actually, I'm oral pathologist. So I would like to ask, like, there are different stages of OSMF. So does this work on all the stages, or like what is the progress of the curcumin?
So there are many clinical studies, more than 12 clinical studies on the effect of curcumin on oral submucosal fibrosis, and most of the studies they have used different types of patients. They have some mild dysplasia plus oral submucosal fibrosis, those stages.
Yes, sir. Actually, this plastic features, like, it's not necessary that in OSMF we can see the dysplastic features.
I understand, but when I showed one slide, the inflammatory cytokines and everything is highly overexpressed in that stage also.
Yes, so that is actually common to all the oral diseases. The inflammation part is common to.
Yes, that is why we are using anti-inflammatory agents.
Okay. Yes.
No,
thank you.
Am I answered your question?
Yeah, actually, I wanted to know, like, with the advancement of the staging, the bands are seen. In the initial stages, it's just the burning sensation, or you can just see the whitish areas in the mouth.
Yes.
So, like, have you seen any, like, in the clinical, like, have you correlated with the clinical cases also? The effect, like, it's on the, I mean, it's, you have worked on the technological part, like on the RNA part, but have you correlated with the clinical part?
Maybe we can talk,
Yes.
Yeah, that is.
Thank you,
nice.
thank you.
Okay. Is everybody ready? It's my great pleasure. Yeah. It's my great pleasure to introduce you the next speaker, Pratap Pati. He is a professor and he is from the Department of Biotechnology, Guru Nanak Dev University, Amritsar in Punjab, which is where I come from too. And his area of interest is medicinal plant biotechnology and agri-biotechnology. Dr. Pratap.
Thank you, Dr. Agarwal. Good morning, all. It's a pleasure to be here, and I thank the organizer for giving this opportunity. And all my previous dialogue speakers have set the ball rolling. And my area is plant sciences. So my focus today will be plant biotechnology, how it is helping in creating or building a healthy society. Building a healthy society and catalyzing the bioeconomy. So when we talk about United Nations new Millennium Developmental Goal, the major emphasis has been given to health, food and nutritional security and environment. And it's rightly so, because COVID has taught us how important is health, and now it is in the top of the global agenda. Similarly, the population is increasing in an alarming rate, and there's a challenge to have access to food and access to nutritional food. So this is another gray area. The third is environment. Environment is, of course, very, very important, whether it is air, whether it is water, whether it is soil, it's very, very important. And these three are inextricably linked. And I'm reminded of the statement of Gro Harlem. This is a very influential lady, two-time Prime Minister of Norway, and she was Director General of World Health Organization. And one of the statements which I quote, you cannot achieve environmental security and human development without addressing the basic issues of health and nutrition. So you can understand how important is health and nutrition. When we talk about health and nutrition, basically the theme of this conference, I'm happy that we've been given a platform to this very important issue. And when we talk about health and nutrition, you cannot undermine the role of plants. They play a very, very important role. So plant as a source of medicine is a very ancient science. I mean, it is mentioned in our scriptures, in our literature, the role of plants as a medicine. What's happening here? Dr. Renu? It's not moving.
मुझे पूछे, मैं बोला मेरे पास मत पूछिए, आप ही लता मैंने, हाँ, उनको बात कर।
Food and fits to the theme of this conference. We have been working on selected plans to the multiplication of the deviation. So that's why we are looking for such plans that are cheaper and so what this we are working at present is the sweet potato, and we might have seen sweet potato which is white in color, but this is a sweet potato which is very rich in the beta carotene. From the color itself, you can see this sweet potato has been developed collaboration with Central Fever Research Institute, and we have been now working on this. So we are providing this in tissue culture. So mass propagation of tissue culture. This is another variety of beta. It is very rich in astrocyte. And you also beta carotene depends on very important different kind of index. So this is very rich in beta carotene and astrocyte. And now we are doing trials in GNU. These two different varieties of the beta carotene rich and antioxidant potato. So they are very nicely from the people that started forming and probably in harvest season in December. And a lot of benefits can take place, a lot of food industry can come into this state and prepare very good and all. And for me, this will be a good breakthrough. So this is another working and this is beta carotene rich cauliflower, and we have been growing this last year. We grow this GNU from our university garden, and we have also with our food
department, research department, we are introducing people. And this is very rich in the technology. And that's why we are working on this. Banana is a very important crop, nutritional powerhouse. But in Punjab, they don't grow banana. It's a very rare site. You see banana. So that's why we thought we should develop a protocol for banana cultivation, commercial banana cultivation. And we are successful in growing banana in Indonesia. And this has been brought into the field. Now, since last 2 years, this is there in the field. This is growing so nicely. They have started bearing fruits. Now, if you visit the university, you can see the banana tree in Punjab, which is very, very rare. So, we are also working in apple. It's a crop which is known for Kashmir and Himachal Pradesh. But this year, we could get fruits of apple in Amritsar in Guru Nanak Dev University agriculture field. And we have been working on developing the rootstock where this will be good for banana. It is said that a banana a day keeps doctor away. So that's why you should understand why we are growing banana. And this banana, this apple you can harvest in June. So in June there is no production of apple, and that's why the cost of apple is around 225 rupees. So we are growing this, which we can harvest in June, and that's why this will be a good breakthrough in giving nutrition to the society. We are growing this rootstock in our tissue culture lab, all these banana, which is very important for low chill apple, and we are successfully developing this technology. Saffron. Saffron is also a medicinal plant. It has aroma. It is a costly spice, and you'll not believe that this is growing in Guru Nanak Dev University. So last year we flowered saffron in our campus, and it was a news. And this saffron, we have been growing it in our tissue culture facility. You can see the palm of saffron, which is growing in our tissue culture facility, because there's a serious problem in getting the palm. So we normally get the palm either from Kashmir or from Iran or in European country. But we have been successfully multiplying in our facility. This is a hugely important medicinal plant. We know this for aroma, costly spice, but a hugely important medicinal plant. So that's why we are growing this in our facility. So because of sort of time, I rushed through my slides. So I'm sorry for that. But I would rather say that plants play a very important role in health and food and nutritional security. With changing trend back to nature, it is important to explore more about plant, enhance their commercial value, and bring them to rational scientific use. With this, I thank all my collaborators, all funding agencies, particularly Dialab, Dr. Renu, Dr. Sunil, and the organizer for giving me this opportunity, and to my parent university, Guru Nanak Dev University, has allowed me to do my research. And at the end, my message to all of you: eat good food and stay healthy. Thank you all.
Questions? Renu said only good questions, not bad ones. Any questions?
There is a student over
somebody
there.
In the back.
Please, can you come over here?
Good morning, sir. Sir, is there any change in the quality of the saffron that you brew here?
This is a good question, but I think the flowering of saffron is itself a breakthrough. But we look into it when we harvest the stigma, because in saffron the stigma is important, and that bears essential phytochemicals. So we will look into it in years to come.
Yes,
Hari, you have to stand up. And here is the mic.
When you grow the plants from 14,000 student, bring them to lab, is the composition of those phytochemicals same or different?
Yes, yes, this is important, and we did analysis. Unfortunately, the student I don't see here, but she will have a poster outside. We analyze this in our laboratory, the field plant and the plant which we collected from natural habitat, and I don't think there is any. So in fact, we could find there are publication already. We could find tissue culture plants have better phytochemicals compared to the field plant. Yes, yes, we have already 2 publications on this.
Any more questions? If not, everybody is very satisfied. Thank you very much. Very, very nice, very elaborate talk.
So we're moving to the last speaker of the session. Our next speaker is Professor Yoshihiro Omiya. He is a prime senior research scientist at the National Institute of Advanced Industrial Science and Technology, Tsukuba, Japan. He is the editor of Journal of Bioluminescence and Chemoluminescence. He is also a chairman of Japan Association of Bioluminescence, Chemoluminescence, and also a past president of International Society of Bioluminescence and Chemoluminescence. So with this, I would like to invite Professor Omiya. Yes. Slide. Desktop, yes, here. Okay. Thank you very much for the. I'm very glad to be here to talk about all my topics. Thank you very much for the organizer to give the good opportunity to talk about all my talk. My talk is very simple to the how to the screening of the bioactive compound based on the mechanical base. And additionally, to important point is high throughput. And mechanical base is very important too. So my talking is the title is a real-time Rush-based reporter system for the nutraceutical screening. So, and firstly, maybe you are not so familiar of bioluminescence. Bioluminescence is a chemical reaction represented as a chemical reaction in vitro, and this is firefly. And extract by hot water and by cold water. And this extract by hot water is luciferin substrate, and cold water remain of the luciferase enzyme. So bioluminescence is a chemical reaction in the enzymatic chemical reaction. And luminescence organisms, there is many type of the bioluminescence organisms. For example, to the right hand is the firefly or beetle type, and then second middle of the jellyfish. This is a luciferin name to the selenterage. Very famous to the jellyfish is contain of the green fluorescence protein. My friend is Dr. Shimomura, discover of the green fluorescence protein. And final is a cypridina luciferase. But luciferin is different but luciferin is oxidized for the oxyluciferin that is excited state return to the basic state to produce to the light. Luciferase the catalyst of this reaction. So luciferin-luciferase reaction is the principle is an oxidation reaction. This reaction is the luciferase powerful reporter enzyme for the gene expression analysis, immunoassay, and imaging. Today, especially it is high performance for screening of the bioactive compounds from natural resource. This is the summary of a characteristic property of a commercialized bioluminescence systems, non-secretion and secretion type. In this time is focusing for the non-secretion type to the quick beetle or something for firefly luciferin beetle systems. But this is, please show what of quick beetle and laserworm is a different color. Same luciferin, but a different color is very important point. In generally, is Promega cooperation to the make to the dual non-secret type luciferase assay. That case is using to the two luciferase. One luciferase is firefly luciferin, and secondary is linear luciferase. Transfer to two vectors expression is depend on the expression level to the promoter region. And after the expression of the two luciferase is Promega system is firstly is put on the firefly luciferin and determine to the target promoter activity, and the secondary is put on the serenterrazine that is the determine for the control. So promoter activity is firefly luciferase activity per linear luciferase activity. However, our group is made to the multi-color luciferase assay systems. This is one-step monitoring of the multiple gene expression with a single substrate. So this is very simple method to different color between luciferase using to green, orange, and red. And the transfection to the three vectors and expression is each color and separated splitting the emission using the optical filter to separation of the three color, and we are determined to the two promoter region per control promoter activity. So characteristic property of three color, that is green, orange, and red. This is very important point, is the color separated with the optical filter, and number of substrates minimum just one, and half-life is similar. This is very important point because of a Promega system is a linear and firefly system is different the half-lives of the protein. That case is watch of the promoter activity. It's not so easy to, but the basic activity is similar. This is another big point is serenterrazine, the linear system is high background, but the firefly system is low background. So a temperature dependency are similar, to very easy to compare to the three gene expression. So additionally, we make the emerald look, E-look. And so we have 4 different color luciferase: green, real green, green, orange, and red color luciferase. Merit is a simultaneously monitor of the multiple gene expression. And the luciferase assay with a single substrate, just only. So real-time monitoring luciferase assay is easy to the separation of the color difference. That is a real-time monitoring luminometer. So in that case is the living cell expresses the beetle luciferase. That case is luciferin in the culture medium to penetrate into the cell and produce the light. Real-time monitoring means to the real-time and determine for the color difference luciferase activity. So in that case, it's very powerful tool for the analysis of a characteristic of the promoter activity. So good target, one good target is the circadian rhythmicity system. So overview of the circadian rhythmicity for human. Adapted of the environment change, orchestral physiological function closely related to the mental activity and critical for the health. So it's important to the circadian system disorder is make to the many disease. So modulation for quality of the life, quality of life. That case is very important to maintain stable circadian rhythm. So that case is circadian rhythmicity system is very interesting system. Mainly is a circadian power 1. That is power 1 is the power 1 is control. It is a 2 gene expression, BMR1 and clock. So mainly the time course is the power 1 part 2 and control. Mainly control to the BMR1 and the clock. BMR1 power is antiphase expression levels. So we make to the 2 color luciferase mice directed to the demonstration coupled expression of the 2 clock gene. One is the green color is the BMR1
and red color is part 2. This show is SCN. SCN is control, center of the control over the circadian systems, lab, some system. So our make to the 2G expression and mice. This mice is the show is very interesting. BMR1 and PA1 is show the antiphase. But different tissue is different, really different to the circadian rhythmicity. So this system, using to this system, we can screen of the some natural compound. So good example of this here, candidate of the circadian modulator, leukocytin, leukocytin, harmony, something for. Today show is one example to here, and the harmine is from the passion fruits. That is in general right hand. This is BMR1 look. Fibroblast line. It is the blue line is a control. But the addition of the harmine to the elongation of the circadian BMR1 expressions. This is analysis of 120 hours. So harmine elongation at the circadian period. In that is very important point because of some cases is jet lag or something. That case is the shortening time to the not so easy to adapt of the circadian rhythm. That case is using the harmine to easy to adapt of the body circadian systems. Another example to here to what the senior. This is, as you know, is the big star of the ashwagandha. This is that case we use. This is preliminary result by Onishi. This is here to control of the black line. We analysis of the 6 days. This is what the senior is have elongation of the BMR1 expression circadian systems, and it's important point. And BMR1 induction, that is very important. More strong, make to strongly is the circadian rhythmicity. That is what the senior and ashwagandha have some potential of the modulator of the circadian systems. So another example to the real monitoring so natural compound for in that case is we're using the IL-6 and IL-10. IL-6 and IL-10 is a green light and the base of the gap DH, red light. So IL-6 is very famous to the inflammatory and IL-10 is anti-inflammatory. So good indicator of the TH1 to TH2 balance. This is we using to the RAW 264.7 cell line and incubation 24 hours put on the Chronos. That is a real-time monitoring of the gene expression. And others, firstly is LPS and or sample luciferin and herpes. So in that case is firstly is the LPS induction of the stress. So in that case is screening of the natural compound. In that case, the glucan from berry. In that case it is just the red light is LPS and the glucan is a nose luminescence. That LPS glucan. That case is really change but not so big influence IL-10 and IL-6. Upper is IL-6, the lower is IL-10. But we're using quercetin. Quercetin is the red light is LPS stress. But quercetin is put on the quercetin is the decrease to the IL-6 activation, not change it to IL-10. So easily to the understanding of the inflammatory and anti-inflammatory. So quercetin have a big potential of anti-stress. So that case is very easily to understanding of the under time course. Another example, LPS depends NFA kappa B was pathway, way pathway. That case is TNF alpha is induction of the some protein, stress protein. In that case, we make to the NF kappa B TK promoter is red light, and the TK promoter is the green light. So in that case, lower figure show is LPS induces the stress. Okay, to depend on the amount of LPS. But plus DEX. DEX is anti-stress. So it's easy to understand the time course and those response of DEX. So we try to the analysis of this system using tamarind seed. Tamarind seed has an anti-stress effect. This is my preliminary result. That is using the tamarind seed extract by the ethanol. Not so purification of the some component, but preliminary results suggest to the tamarind seed have the potential of the anti-stress effect. So finally, is a summary of my work. Real-time risk-based reporter system is a powerful tool for nutraceutical screening by active compound from natural resource. In this system, we can easily simultaneously evaluate the dose dependency and effective time course and toxicity of candidate. Luminescence analysis, now we can use it to 96 well plate machine. This equipment very easy to high throughput analysis of the compound. So I hope to using this system to Indian side to the screening of the good natural compound from the natural resource. Thank you so much.
Thank you, Professor Omiya. The talk is open for any comments, any questions. Anybody wants to ask any questions to Professor Omiya? No? So if not, then I would like to thank again Professor Omiya, and I would like to thank my co-chair, Professor Agrawal, Professor Katyal, and all the 6 speakers for excellent talk. And with this, we come to the end of the first session picnic. Thank you so much.
So thank you, speakers, for your informative lectures and the valuable time. Now I request chairpersons to give the memento to the invited speakers, Dr. Sunil Kaul, Professor B.B. Agarwal, and Professor Anju Katyal. I request Dr. Renu Wadhwa to come upon and receive a memento. Just 2 minutes, sir. Just 2 minutes. So I request Dr. Renu Wadhwa first. Sir, photograph, ma'am, photograph. Next we have Professor Durai Sundar. Thank you, sir. Next we have Dr. Fayaz Malik. Next, Dr. Ajay Kumar. Next in line, we have a Professor Pratap K. Pati. And the next invited speaker is Dr. Yoshihiro Omaya. Thank you all the invited speakers for telling the different nutraceutical interventions and the therapeutic potentials. I want to thank on the Department of Zoology, on the behalf of all Department of Zoology. Thank you all. Heritage Walk के लिए सुनो ममता गेस्ट कौन है देखो है So I request Dr. Professor Bhushan Patwardhan to come up on the stage to felicitate chairpersons for the session 1. Sir, come at front. Dr. Sunil Kaul. Professor B.B. Agarwal. And Anju ma'am thi na. And Professor Anju Katyal. Sorry, sir, and thank you, ma'am. Can we have a one photograph with all the chairmen and the invited speakers? Ma'am, we have another photograph session just after this.
Just count 5 from now. 1, 2, 3, 4, and 5. Please take pictures. Quick. Okay, picture time. Right. 1, 2, and 3. Here we go. Thank you very much.
Now we have a next session for the Heritage Walk. Everyone, please settle down fast. The revolutionary leader was born in 1907 in Kharagpur district of Punjab provinces of British India. Is remembered for his intellectual contributions which is now Singh jail diary, Chabri, and old lanterns that was an only sources of the light. There is a photo of bicycle watch. So we give a tribute to the Shahid Bhagat Singh. Heritage Walk is the Shahid Bhagat Singh Smarak at the University of the Delhi, commencement of the Bhagat Singh Subte and the Rajastri by paying homage to Bhagat Singh in the chamber located in the basement of the Rice Lodge of the university where he was imprisoned. An exhibiting collaboration of notes on freedom fighters 1:30 p.m. Those who are interested, give me a name at least. Thank you. Now we are going for a tea break, and after that we'll be all going to assemble for a group photograph. Thank you.
आ रहा हूँ सर, आ रहा हूँ।
ये देखो अभी क्या नहीं है भाई पीछे कट कर रहे हो ऊपर बीच की डाल रहे हो बैकअप डाला हुआ है ना इसलिए आ रहा है ठीक है 20 सेकंड दिया भाई दोनों दोनों बंद रहो दोनों ऊपर नीचे बंद रहो जो पहले वाला बंद था ना अब उसके ऊपर वाला बंद हो गया बैकअप निकालो बाहर जाके बैठ गए तो तुमने फोन करूंगा ये फोन कर दिया होगा भैया जगह बढ़ा दिया इसका आ गया नहीं आया जब मैं बैकअप बढ़ाता हूँ जा आप ठीक होगा ठीक है फिर खराब कर दिया फिर गया हमेशा सब दे रहे हैं हमें कोई देना तो देगा चाहिए आओ आप बताना ये ऊपर से गई हुई है देख ऊपर से आ गया देख देख ये ना दोनों की भाई नीचे कर रही है सुन 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 पहले ना भैया पहले लिखा है ना अब सिग्नल चेक कर ले नहीं ये है दो दो लाइन आ रही है दो के ऊपर से नीचे दो आ गए नीचे से नीचे गए हाँ अरे हाँ भाई सिग्नल दिख रही है क्या लाइट दिख रही है क्या लाइट दिख रही है बहुत ही ऐसे से चल रही है भाई जितनी चल रही है भाई वो तेज चल रही है वो धीरे चल रही है तो भी सिग्नल ही अरे वो तो आ गई ना हाँ ये वाला तो आ गया अभी सीधी चल रही ये आ गया अभी सीधी चल रही सीधी चल रही सीधी चल रही है सीधी चल रही है नहीं है काम चालू होने से पहले चालू चेक कराने के लिए बदल बदल चलो हाँ एक आ गई अब सीधी चल अंदर अब सीधी चल अंदर एक आ गई एक बार आ गया आ गई अरे आई थी आई थी आई थी आई थी हाँ ठीक है रुक जाओ अब मुझे बैकअप देखने दे बैकअप देख रहा हूँ ठीक है दोनों ठीक है लो जान दो एक सर एक
एक HDMI के दो ये इसको शेयर करने के लिए क्या दबाते हैं? F10? हाँ F10 दबाते हैं। हाँ देखो 4 लोग आ रहे हैं, 25 चीजें मुझे तरीके बता दे रहे हैं, तो मैं क्या करूँ? कनेक्ट नहीं कर रहा हूँ, मैं चेक कर रहा था कि उससे होता है या नहीं। कंप्यूटर आपका हाल चल रहा किसी और का अच्छा कंप्यूटर मांगू क्या? हाँ एक मिनट फोल्डर S1A मुझे वन ड्राइव का कुछ नहीं पता है।
छोटी वाली
ये तो
मुझे चाहिए पानी।
चश्मा कॉपी करके
तो मैं पानी पी के आ रहा हूँ।
ये है ना।
इसमें टेस्ट कर दूंगा कहाँ गया फोल्डर
यहाँ
सॉफ्टवेयर डेट कर देता हूँ ऊपर आ जाएगा है ना डेट है
ठीक है।
नेशनल सॉन्ग के लिए तुम बोल रही हो ना?
इसमें टेस्ट करना ना।
हाँ
अबे यार
वन ड्राइव नहीं बोला था गूगल ड्राइव में बोला था या कुछ या लोग संग में जो भी
आई हैव नो आइडिया।
कोई दिक्कत नहीं अब बीपीटी नॉन टारगेटिंग मेटाबॉलिक रिक्वेस्ट ना ठीक है और ये निकाल दो
ड्राइव निकाल दो फिर ठीक है।
ध्यान रखना
एक क्लास का एक तो दो क्लास और चाहिए ना
मक्खन मार रहे हो आप।
सर को है ना ये प्लास्टिक से दिक्कत है इसीलिए बॉटल चिकन नहीं लगा रखी या तो बहुत नीचे हो गई क्यों पड़ा है I have heels पहन के हाँ बट वो ऊपर है तुम कितनी ऊंची हो वो हील थोड़ी पहन रखी थी मैंने अच्छा ठीक है अब ये दे दूं बॉटल यहाँ रखी पहन पड़े छोड़े पड़े तो तुम्हारा सीक्वेंस आ गई ये माय गॉड है वेरी गुड ओनली तो दिख नहीं रहा है कोई नहीं अरे ये नहीं दिख रहा वेरी गुड Hold the everyone seated, hold it. Hold the you are seated.
Everybody is requested to please be seated in their places. A very good afternoon to all the dignitaries, scientists, and colleagues. On behalf of Department of Zoology, University of Delhi, we are honored to welcome all the speakers and participants from all over the world to our 5th International Conference on Neuroceuticals and Chronic Diseases. Incd conference is designed to explore the scientific basis for the pharmaceuticals and nutraceuticals for cancer and other chronic diseases. The conference is sponsored by UGC Special Assistance Program on commemorating 100 years of University of Delhi and 75 years of Department of Zoology. The University of Delhi is a premier institute of the country with distinguished legacy and an international acclaim for higher standards. It was established in 1922 with motto Nishtha Dhriti Satyam, dedication, steadfastness, and truth. The university is completing 100 years, and it is no less a momentous occasion to reflect upon our institutional success, achievements, and glory to Department of Zoology completing 75 years of excellence. Following its inception in 1947, under the leadership of Professor M.L. Bhatia, the Department of Zoology has been recognized as the Center for Advanced Studies since 1963 and has continued to maintain the status till date. Topics such as chronic diseases, especially cancer, and others such as diabetes, obesity, mental disorders, arthritis, cardiovascular, pulmonary, and infectious diseases will be covered. Others such as epigenetics, biofilms, microbiome, and omics, and the changes by nutraceuticals will also thrown light upon. I now request our head of the department, Professor Reena Chakrabarti, to accompany Honorable Vice Chancellor, Professor Yogesh Singh, our guest of honor, Professor Bhushan Patwardhan, Chairperson, Professor Bharat Bhushan Agarwal, Professor Daman Saluja, Chairperson, Research Council, Professor Ajay Kunumakar, Secretary NCD, Professor Alok Chandra Bharti, and Professor Anju Srivastava, Organizing Secretary of the conference, to come upon the dais. I request our PhD students to welcome our dignitaries by presenting them bouquets. Now, we would like to request our PhD scholars and MSc students to recite the national song.
Ayo.
Vande Mataram. Vande Mataram. Sujalam Sufalam Malayaj Shitalam Shashashamalam Mataram Vande Mataram Shubha Jyotsnam Pulekitayamini Kusumita Dromedale Shobhini Suhasini Sumadure Bhashini Sukhidam Varidam Mataram Vande Mataram Vande Mataram.
Thank you, everyone. To make this event a blessed one, I request our head of the department, Professor Reena Chakrabarti, to escort our Vice Chancellor, Sir, and our Chief Guest, Professor Bhushan Patwardhan, Sir, and other dignitaries to kindle the lamp and inaugurate the conference by lighting the lamp. Now, I would like to request Professor Anju Srivastava for the word of welcome.
So good afternoon, everyone. Honorable Vice Chancellor, Professor Yogesh, Honorable Chief Guest, Professor Bhushan Patwardhan ji, respected Chairperson, Professor Bharat B. Agarwal, our Head of the Department and Convener for this conference, Professor Reena Chakrabarti, all the distinguished dignitaries on the dais, all the delegates, all administrators, and their students. On behalf of the organizing committee of Incd 2022, we welcome you all to the 5th International Conference on Nutraceutical and Chronic Diseases. It's a matter of great privilege and pride that this conference themed Pharmaceuticals and Nutraceuticals for Cancer and Other Chronic Diseases is being organized by Department of Zoology, University of Delhi. It is also highly satisfying that the conference is designed and executed in physical mode and thus marks the resumption of routine activities post-COVID time. We congratulate one and all for their enthusiastic participation in this event that will drive it to its success. With a lot of uncertainty looming around, it was a challenge conceiving a physical meeting a year ago. But with God's grace and remarkable teamwork, we could overcome the hurdles of gathering together in physical meeting, and we are highly indebted to all for their kind gesture of accepting our invitation in this time of great uncertainty. This year marks the memorable milestone for our university as it completes 100 years and the department attains 75 years of academic excellence. On this momentous occasion, we celebrate our people who are instrumental in attaining this landmark. We invite all the guests and delegates to share our happiness over the achievements during this meeting. The event is combined effort of three different societies: Society for Nutraceutical and Chronic Diseases, Society for Translational and Cancer Research, and our own society from Department Evolvera. It's a society of students, and their enthusiastic participation is very, very motivating. This meeting aims to bring together distinguished scientists, clinicians, physicians, nutritionists, allopath, Ayurvedic, homeopathic, Unani, Siddha, traditional medicine practitioners, young researchers, corporates from different within country and abroad to discuss and deliberate trends and future prospects in basic and translational nutraceutical research and treatment. The conference will provide an interactive platform for exchange of new ideas and recent development with peers in different domains. We are sure that this meeting will provide ample opportunity for networking among the researchers from various countries. Further, attendees will have an enriching experience to interact with the veterans, renowned scientists on one hand, and budding teachers in the field through a series of over 70 scientific lectures in different academic sessions, including 3 public lectures, 3 plenary sessions, 12 scientific sessions, a panel discussion, 6 oral presentation of special highlights, and 3 poster presentation sessions for young researchers. One session is exclusively dedicated to breast cancer, a leading malignancy in Indian women. Furthermore, national and international delegates will have an exciting indulgence to the cosmopolitan culture and tradition of North India while it is in festive mode. The proposed conference is designed to explore the scientific basis for the use of pharmaceuticals and nutraceuticals for cancer and other chronic diseases. Nutraceuticals derived from spices, dietary agents, medicinal plants will be included. Chronic diseases such as cancer, neurological and neuromuscular disorders, metabolic disorders, immunological disorders, and emerging infectious diseases will be covered. Topics such as emerging anti-cancer phytochemical, their molecular targets and drug delivery, Ayush therapies, and frontier areas of nutraceutical research will also be addressed. The entire academic extravaganza would not have been possible without the strong financial support from University of Delhi and other government funding agencies such as DST, SERB, DBT, DRDO, CSIR, INSA, Ayush, and DSIR. A timely support from corporate sponsors like SAMI, BD, Biorad, Insights, VACO, and others is thankfully acknowledged. Generous contribution from friends like Narula Research, USA, IMM Germany were a lifesaver for the conference. The NCD is indebted to the offers of Professor Acharan Narula to support the current and also the upcoming INCD conferences. With this, we thank our collaborating institution as well, particularly in MAS, ACBR, and Miranda House. They are instrumental in helping wholeheartedly in every endeavor of the conference. We extend our heartfelt gratitude to all the people of the organizing committee, volunteers from Department of Zoology who have contributed to make the event a memorable one. We express our sincere gratitude to all the participants for their active participation in this meeting, despite of different logistic hurdles caused by pandemic, and for encouraging their colleagues and students to be part of this conference. We are sure your participation in conference will be enriching and rewarding experience. Finally, we hope that your stay in our modest accommodation provided in different guest houses will be comfortable. Any inadvertent inconvenience caused is deeply regretted, and it is on us. Once again, we welcome all of you from everywhere. The organizing committee members have tried their level best to put together excellent scientific program, social and cultural program. We hope that these 3 days will make a memorable experience. Thank you very much for listening, and let us hope that these 3 days are going to be very, very rewarding. So on behalf of the organizing committee, I welcome everybody.
Thank you, ma'am. I now request our regarded head of the department, Professor Reena Chakrabarti, to say a few words.
Thank you. Respected Vice Chancellor, sir, Honorable Professor Bhushan Patwan ji, the Chairman NAC, Professor Ajay Kumar ji from IIT Guwahati, Madam Professor Daman Saluja, Chairperson of Research Council, University of Delhi, esteemed delegates, the soul of the conference, the members of the organizing committee, the esteemed faculty members, and beloved students. I am feeling thrilled and extremely privileged to welcome you in the 5th International Conference on Nutraceuticals and Chronic Diseases, organized by Department of Zoology, University of Delhi. Department of Zoology has completed glorious 75 years. Significantly, our nation is also celebrating this time 75 years of independence, the great Amrit Mahotsav. The Department of Zoology during the years has made significant contribution in teaching and generating knowledge in various aspects of animal sciences. In view of its great impact on national education vis-a-vis nation building, the University Grand Commission declared it as the Center of Advanced Study for Zoology as early as in 1963. The University of Delhi has also completed its 100 years. The centenary year is being celebrated with due gaiety under the stewardship of our family head, Honorable Vice Chancellor, sir. We are indeed fortunate to have such an able administrator as well as great scholar like him as our Vice Chancellor in this Amrit Kal. Professionally, he is a software engineer whose research contributions have earned high H-index and citation index value. It is a matter of great pride that a person of the stature of Professor Patwardhan is gracing the occasion as the chief guest. We welcome you, sir. We appreciate the glorious presence of Professor Ajay Kumar ji, who has taken pain to travel from Assam. We heartily welcome you. Madam Saluja is one of us. She is our very helpful friend. We extend our cordial welcome to you, madam. I congratulate the organizing committee and its esteemed members to organize so meticulously the international conference for the 5th time. I wish fruitful scientific discussion and happy stay. Wishing all of you my belated Dasara greetings. Shubha Vijaya, Namaskar.
Thank you, ma'am. Thank you, ma'am. I now request Professor Daman Saluja to address the conference.
Good afternoon, everybody. It's almost noon. Respected Vice Chancellor, sir, Professor Bhushan Patwar, Professor B.B. Agarwal, my colleagues, friends from university and outside, and my dear students. I, on behalf of University of Delhi and in my own capacity as the chairperson of Research Council, welcome you all to the 5th International Conference on Nutraceuticals and Chronic Diseases, organized by the Department of Zoology to mark their 75th anniversary, as well as to mark the 100 years of glorious journey of University of Delhi. The University of Delhi really does not need any introduction. And as has been told my previous colleagues, this has been and always had been and still continues to be a prime university and it's like a dream university for every student in India to come and study. So, and I'm sure under the leadership of our current Vice Chancellor, Professor Yogesh Singh Ji, it will go into further heights as he is really working very hard to bring the levels of research and teaching to a better level and high ranking in IQAC. So I think this conference has come up at a very, very right time. As we observed in COVID time, many of the people had gone back to the natural, therapy for COVID, because we really didn't know much about the disease, the pathogen, as well as the medicine was not available and diagnostic was poor. So people realized once again the importance of the naturopathy in our country in a very, very overwhelming way, I would say. I heard and I was reading a lot about several plant decoctions being taken by people all over India, and that really helped. And I think this conference rejuvenates the importance of the natural products in our society, which have always been emphasized by various studies and since ages, since olden times, the Ayurveda, the Unani and Siddha and all that, homeopathic. So once again, we are here to relook into those medicines. And our scientific galaxy that is present here and who will be over the next 3 days will be talking about various work of they have conducted in their labs and in their institutes will emphasize how they have proven the effect of these natural compounds using modern tools and techniques. That is very, very important because people had stopped using these natural products because they thought that they have no scientific basis. So now this, over the next 3 days, as you must have seen the program, over the next 3 days, you are going to see how they have gone ahead and proven all these concepts once again. And you have to also remember that all the chronic diseases, although the focus here is more on cancer, but most of the chronic diseases are multigenic. And if you say multigenic, that means multiple pathways will be affected. And therefore, a single drug will not be effective against these chronic diseases. For management of this, all the chronic diseases, we need multiple drugs or naturopathy, which will help in regulating or managing several pathways simultaneously. In fact, if you look into the history of many modern medicines in allopathic also, you will see that they have origin from the natural products. They have been modified or they have been synthesized in the laboratories or in the factories or in the pharmaceutical companies, but they have origin from the plants. And this gives us a big hope that by rejuvenating our own resources, we will be able to come out with naturopathy as one of the leading drug system or medicine system for management of chronic diseases. I'm sure that over the next 3 days will be very, very fruitful in all these areas, and you will have a wonderful scientific treat listening to the galaxy of eminent scientists working with natural compounds on chronic diseases. I'm sure that this conference will help in bringing all the scientists and experts in the field to come under one umbrella and discuss their valuable discoveries, and establish new collaborations. I wish good luck to all the organizers, speakers, delegates for their success in this conference. Thank you very much.
Thank you, ma'am. Now, I would like to request Professor Bharat Bhushan Agarwal for introductory remarks.
Dear friends, ladies and gentlemen, first of all, I would like to welcome all of you to this conference on nutraceutical. And I just want to add a few words. If it was not for Dr. Alok Bharati and Anju Shrivastava, we will not be here. So I want to thank both of you, both of them, for organizing this conference. And they've done an incredible job, tremendous job. Second thing that I want to add is that we all know that it is a mother who brings us in this world. If it was not for the mother, we will not be here. And then it is a mother nature that takes care of us after we are born. Question is, what is there in mother nature that takes care of us? And that is what this conference is about, that what is there in various plants that helps us to live, to survive in this world. And it just so happened that I happened to discover a molecule that is supposed to cure cancer. And that molecule some of you may have heard of, called tumor necrosis factor. And that tumor necrosis factor turned out to be tumor promoting factor. As a result of it, it turned out to be a primary mediator of inflammation. And the blocker of that particular molecule now has a market over $50 billion a year for last 30 years. And mind you, we discovered haldi, turmeric, curcuma longa can do just that, block that molecule. And that was our introduction to the natural products. And we've been working a lot on the natural products. And a number of people have worked with us over the years. And it is unbelievable. And this conference is a perfect format. And this is what we decided. My name is Bharat. And I am a product of the University of Delhi. I was just remembering myself that first time I came was 1968. That probably some of you may not have even born at that time. That's when I first time arrived over here and I started my career right here. And after finishing my undergraduate, I moved to Banaras and then I ended up University of California, Berkeley. And then from there UCSF and then company Genentech, where we discovered TNF, and then we moved to MD Anderson Cancer Center, and that's where we got into all the natural products, that how they work for cancer and all the various diseases. And mind you, there is no limit to all this. And I think that what you are hearing today is tremendous. And that insight that everybody is providing what this natural products can do for us. And I want to thank all of you for being here, for providing all the information that you know. With that, I thank you. Bye-bye.
Thank you very much, sir. Without any further ado, we would now like to request our dignitaries present on the dais to launch our abstract book.
Now, I would like to request our Honorable Vice Chancellor, Sir, to felicitate our Chief Guest, Professor Bhushan Patwardhan, by presenting them with shawl.
I request Professor Daman Saluja to present a memento to our Chief Guest, Professor Bhushan Patwardhan, sir.
Now, I request Professor Bharat Bhushan Agarwal to felicitate our Vice Chancellor, Professor Yogesh Singh, by presenting them with shawl.
And I request Professor Reena Chakrabarti to present a bouquet to our Honorable Vice Chancellor, sir.
We would now like to request Professor Bhushan Patwardhan to address the conference.
Thank you, organizers, for inviting me to witness this historical event, I would say, marking 100 years of this great university and 75 years of this great department. I'm so happy here today when we were just sitting inside, and Vice Chancellor Professor Yogesh Singh, whom I know as a great leader, told the head of the department that please call all the new recruits, And this was like music to my ears. Because wherever I go, I get to hear that there are so many vacancies in universities is not advertising, the faculty positions are not filling up. And congratulations, Professor Singh, for this great achievement. And that also I liked his gesture, He invited all the young faculty members, let them introduce themselves, And this shows the quality of a great leader. I'm happy that University of Delhi is in good hands. I also want to thank one who I have held in great esteem. Many of us know him as Spice Man. Many of us know him as Haldi Man, Turmeric Man. But I also know him as TNF Man, because very few would know that TNF, which has become such a crucial marker for all of us during research in immune inflammation or in cancer, he is the one who actually very, very early initiated it. So thank you, Professor Bharat Bhushan Agarwal. And I was telling him that my name is incorporated in him. So I'm happy. His name is Bharat because he was born in the free India. Thank you, Professor Alok Bharati and your entire team. So meticulously organized conference, very, very carefully done, and a galaxy of speakers. The whole arrangement of sessions and selection of speakers has been really very impressive. And your colleagues, Dr. Reena, Dr. Daman, Dr. Anju, and Dr. Ajay Kumar, who has come from IIT, who also happens to be Professor Agarwal's student, and of course my colleague from Pune University, Professor Kalpana Pai. All of you, all friends, all researchers, especially all young students, And especially for students, I would say that you are really fortunate who are studying in this university, because this university, every Indian aspires to come at some point or other to this great university. Also, congratulations to the comparers, they are doing a great job. Thank you. We are running little late, so I'm going to save some time so that we finish in good time. May I have my presentation? It's on. Okay, so then I would stop here and thank you very much for inviting me and look forward for more interactions with you. Namaskar, Jai Hind.
Thank you very much, sir. We would like to request our Honorable Vice Chancellor, sir, to say a few words.
Good morning to all of you. Outstanding and distinguished academician, Professor Bhushan Patwardhan ji, Professor Bharat Agarwal, Chairperson INCD 2022, Professor Daman Saluja, Chairperson Research Council, University of Delhi, Professor Reena Chakravarti, Head of Zoology Department, University of Delhi, Professor Ajay Kumar, Executive Secretary of the conference, Professor Anju Srivastava, Organizing Secretary, Professor Alok Chandra Bharati, who is also the Organizing Secretary, Deans, Academics, Professor Ratnabali, Deans and Head of the Departments of University of Delhi, respected speakers, scholars, delegates, invited guests, faculty members, my dear friends and students, good morning again. Very good morning to all of you. Delhi University has completed 100 years. Congratulations to all of you. Three good things have happened. Delhi University has completed 100 years. India has completed 75 years of its independence, and Department of Zoology has also completed 75 years. India has done extremely well in last 75 years. India will do very well in another 25 years. Department of Zoology has also done very well in last 75 years and produced excellent scientists, faculties, researchers to the world. When Professor Bharat said he studied from University of Delhi and then for post-graduation he went to other university. And this is your university, sir. I would like to welcome on behalf of University of Delhi in the centenary year of University of Delhi. Now coming to the conference, I would like to thank Department of Zoology to organize this conference on a topic which is very, very important and relevant. Why I am saying it is important and relevant? Because after COVID, many things have changed. And I am not from that field. And day before yesterday, I cancelled my program to come to this conference because I had to attend other meetings. So I was very confident, I don't have to go. Why should I worry about the details of the conference? But then fortunately, my program was cancelled and I decided to come to this conference. And yesterday at about 9 or 9:30, I was worried, oh, tomorrow I have to speak. I have to speak on a topic, on an area which is totally unknown to me. I don't have any idea, but I do understand, then, I thought when we say this nutraceuticals, what could be the simple meaning of nutraceuticals? Because this word was very, very unknown to me. I immediately went to the Google and tried to understand the subject, the word. And, then I got very simple meaning of this word nutraceuticals, and which is using food as medicine. We go, oh, it is very easy now. Food as a medicine we use every day. This is very important and very useful and very meaningful. But in the science, we use very scientific words, rightly so also, but sometimes for a common person, those words are very difficult words. When we say like chronic disease is fine. I understand the meaning of chronic disease, and so using food as medicine for chronic disease. So I could understand the theme of the conference, and I am confidently I can say that your topic is very important and very relevant after COVID-19 era, after COVID-19 world. And whenever we say anything, food as a medicine, immediately क्या शब्द आएगा हमारे ध्यान में? We come to Ayurveda. Ayurveda, and ma'am has also said the Ayurveda, and the meaning of Ayurveda is what? It is आयु को बढ़ाने वाला वेद. आयु को बढ़ाने वाला वेद, which is Ayurveda. And it means what? The medicine, the food which we take, if the food can act as a medicine for us, when to take, how to take, how much to take, these things are very, very important. And in this modern world, and particularly in this 21st century, we are very, very reluctant about it. घर में मम्मी कहती थी रात को दही मत खाओ, दोपहर को खा लो, रात को नहीं खाना. अब हम तो रात को ही घर जाते हैं, रात को ही दही खाते हैं. So it means what? We are taking food. Dahi is a medicine, but it may not be useful because we are not taking at an appropriate time. That's why when to take, how to take, how much to take, these are also very, very important aspects. And if we can, like you all are scientists of this area, if we can create some medicine out of this, then that will be very good. I can take that medicine and then I'm confident that I have taken it and the medicine will take care of many things. What we do in case of allopathy medicines, we are not worried about the ingredients of the medicine. And I have a doctor who is my friend. He said, I don't attend those patients who are interested to know the ingredients of my medicine. They should believe me. They should believe me, they should respect me, they should understand, but this belief is very, very important. Otherwise, I don't give medicine to Google patients. I don't treat them. But the same thing which an allopathy doctor is saying, in Ayurveda this is inbuilt. कि पहले हमको भरोसा होगा. क्या लेना है, कैसे लेना है, when to take, how to take. That's why this is very, very important. And the two other words which our Dean research, she has said about the spices and herbs. Both are very, very important. When we say the Ayurveda, these two things also comes in my mind, that herbs and spices. And India was famous for spices exports in ancient times. I really don't know, my dear young students. In 2021, India exported 1.76 million tons of spices. And India is the world's largest spices producer, also a consumer, and also exporter. So we produce also, we consume also, and we export also. सब में हम सबसे ज़्यादा हैं. हैं ही हम 131 करोड़ तो ज़्यादा तो वैसे ही हो जाएगा हर काम हमारा. Because size is also important. And when we say the Indian economy is now 5th in the world, but actually we are only 3% of that economy. We are better than British economy, but our per capita income is only $2,000, $2,050 per year, and there is more than $10,000 or $12,000 per year, or even more than that. So point here is, as a researcher, it's our responsibility to take lead and create something new for the country. And these are the areas where we have brand. Our companies have done very well. India has reputation worldwide for the export of spices and herbs. If we create something out of that, but what is happening in our Indian university system, our research is confined to extension work. It is confined to redefinition of new things and reinterpretation of the existing ideas. My dear students, you have to work here for another 30-40 years in this area. Please create. Your objective should be, as Professor was proudly saying, and rightly so, I have created this for cancer. Yes, in our lifetime, we should be able to, say something with confidence that I have created this and I will help humanity in this way. That should be the objective of the research. Publication of research papers are fine. Impact factors are also very good. But that is only a part of it. That is not the whole journey. If you satisfy yourself after publication of a good research paper, maybe very good for you, maybe good for promotion, wo aap bani jaoge na. Those who have joined this university system as assistant professor, you will be professor one day, whether you work or. But point is not that. Teachers are institutions. You have to create your own world, because in this system, if you do not work or hardly work, then also you can survive. But if you work very hard, create something, the respect, affection and influence, your influence on your students, that is the credibility of a teacher. That my dear teachers also, you should please think about it, that I should be a credible teacher amongst my students. And respect in the eyes of the student is ultimate thing in the life of a teacher. That's why we have to reset our goals, and in such conference, if they can help us to do that, that is the purpose of organizing those conferences. We have personalities like Professor Bhushan Patwardhan ji and Professor Bharat and our own professors also. They have done something very significant in their lifetime. We have to learn lessons and try to. But now replication may not help us. We have to work further, because India, if we have to go from this $3 trillion economy to 10 trillion, and then we want to have $18 trillion economy. Reason is what? We are 18% of the world population. So our contribution should be 18% only, not more than that. We don't want anything more than that. We are 18% of the world population. Our GDP size should be 18% of the world GDP. Presently, it is $100 trillion. R is only $3 trillion. That's why we see poverty in our country. And this poverty can be eradicated. India can become a very prosperous nation with the help of research only. And these areas where lots of potential, and we have brand India in that. If we can create our own word in that, that is the purpose of working in this research area. And the scholars and the university like Delhi, we have to create some success stories for that. My good wishes to all the researchers for that also. Now, in the times of ancient time, when Ayurveda, Ayurveda was very powerful in the ancient India and in ancient world also, many people used to have 100 years life, 120 years life, 130 years life also, 140 years of life also. But now, with the help of this modern medicines and modern lifestyle, our life is what now? We hardly find a person who is more than 100 years. 90 में हाल खराब हो जाता है आजकल, हो जाता है ना? But average age is now in India is 70 years. Average age, yes, good, that's fine. But what is happening, and particularly in Delhi, our air is polluted, water is contaminated, food is also contaminated. We are using lots of fertilizers and pesticides. So so the challenge is what? We are trying to address those issues also. I'm not saying that, but as a person, if I have to do something for myself and for my family, then I have to increase the immunity level of my system. How can we? This research and such medicines can help us, because they are coming from plants, they are coming from vegetables. If food as a medicine, if I can enhance my immunity level, then without taking those modern medicines, I can help myself and I can help also my family. When we talk about the chronic disease, cancer is तो बहुत बड़ी बात है, वो डर लगता है कि हुआ तो गए, ऐसा लगता है. Very true also in most of the cases. I lost my father, when cancer was detected. He was a professor of mathematics and very active person. One fine day, he detected cancer, and doctor said that was small cell carcinoma. ये तो लाइलाज है, and he has only 10 or 20 days. We all were shocked. How can it is possible? One fine day, person was very active, although he was having some kidney issues, but kidney issues was also due to cancer. Nobody could realize that. And then doctor said, when allopathy says something, we have to take it seriously. They say he has life of 10, 20 days. And he died after 18 days. The point is, those are very, very big things. But, the accelerator for that is diabetics, that is blood pressure. They have also become very chronic disease nowadays. And if we can help there, a book has come in the market. I don't know how many of you have read that book. Homo Deus, A Brief History of Tomorrow, written by Yuval Noah Harari. He said in 2012, 1.5 million people died because of diabetes. And sugar is more, he said sugar is more dangerous than gunpowder. And 40% who have died from COVID-19, 40% of the people who died due to COVID-19 had diabetes. This is another research. And, we have two challenges now in 21st century. Very, very serious challenges are challenges. I should not say serious. We should take it positively. So our research aim should be to overcome this old age and make people positive and happy. He said, author said, our objective should be to enhance this age and make people happy and what? Make people happy, productive, because if someone is not productive, someone can't be happy. These things are related to each other. So in 21st century, we have almost double our life expectancy from 40 to 70 years. And in 21st century, research scientists, we all are very confident that they will further double this age. And this will go up to 70 to maybe 140 or 150. Kya hoga tab? 140, if I have to live for 140 years, my total thought process will be very different. I should be valuable. I should be useful for another 40, 50 years. It means, and then another is, how can we make people happy? When we are living with an average age of 70, हम में से बहुत सारे खुश हैं ही नहीं. आप में से कितने लोग खुश हैं? बहुत थोड़े ना? कोई बात नहीं, हैं? क्योंकि किसी ने बहुत अच्छा कहा है कि सबको गिला है कि हमको कम मिला है. सबको गिला है कि हमको कम मिला है. सबको गिला है कि हमको कम मिला है. अरे ये तो सोचो आप जितना कितनों को मिला है. आप जितना कितनों को मिला है. And this artificial intelligence, biotechnology and advancement in Ayurveda will change the world significantly. I am very confident, and your area will be a leading area in this direction. The one is digital world, IT, software. Second area, which is these medicines from plants. Food is medicine, because if both the areas will go together, then and only then humanity will be benefited in the larger way. So I'm sure that you will learn new things on all these 3 days, and the sharing of research would help to develop more nutraceutical based products for the prevention and treatment of chronic disease. So my dear students, think about it. If you are here, don't copy. Those days are also over. Now, please don't cut, copy, paste. In the research, please, and come out of this, develop your own expression, because we Indian students, because our mother language is not English. So sometimes we are hesitant about many things. So develop your own expression, but decide that we will not copy anything from anywhere. Try to find some, useful problem, work in your own way, and promise to yourself that before leaving this world, I would like to create something for the humanity in whatever way you can create. Then and only then, the purpose. And in the centenary year of University of Delhi, University wants this type of thing from every dear students. My good wishes to conference organizers, and I'm thankful, and I again welcome you all in University of Delhi. Thank you very much.
Thank you so much, sir. I now request Professor Reena Chakravarti to facilitate our Honorable Vice Chancellor by presenting him with the shawl. Now, we would request Professor Alok Chandra Bharti to deliver the vote of appreciation.
Thank you. Good afternoon. We started with good morning. Now we have reached to good afternoon. And this is a time we have to wind up the inaugural session. It is customary to first of all pay our thanks to the most important people, particularly the chief guest and the guest of honor, or the person who is presiding, our vice chancellor. But I think I want to break this trend, because this conference, if you remember, part of the people who are actually in designing this conference, they remember we conceived this conference 1 year back. And somewhere in July or August of 2021, it was not certain that we will have another wave or another kind of activity where everybody will be further confined. So I will appreciate and I'll express my thanks to each and every one who has accepted our invitation and thought that we will have this conference in a physical mode in October 2022. So I think it's a matter of great applause, and I'll thank everyone for accepting our invitation. And certainly, I will also bring a very short story to you, because we were looking for people in nutraceuticals to make a list of invited or distinguished speakers. But at that time, we never had thought that we will call any big, person for as a chief guest. So one fine morning, we were looking who are the big people who have done great deal of work in the area of nutraceutical studies and taking it to the clinical part. Because, every here and there you see in different universities and institutions, people are working, preliminary work and trying to take it to the clinical world. And here we found a person who have actually taken this nutraceutical work right from the lab to the clinical nature, and then he had also established an institution who's working into the scientific basis of Ayurveda. And it was none other than Professor Bhushan Patwardhan. And it's a great applause to him that in one go, he actually accepted being a technical person and being a very highly placed in administration. He still had no hesitation. He has immediately accepted it, that I will be part of this conference. I'm greatly indebted to him. Another very important person was our, vice chancellor. But to reach to the vice chancellor, there are different gates. And one of the most important gate was our dean academics. I think I expressed my sincere thanks to her. She made our way so smooth so that we can have an approach to our honorable and very dear Professor Vice Chancellor, Professor Yogesh Singh. It has been, of course, you always know when a person is placed in a very high chair, how much responsibility that person bears. And you don't know exactly what is going to happen next day. So I have always heard that our Professor Vice Chancellor, sir, he was very positive to come to this meeting. So I called to his office one day, okay, he is coming. Next day, okay, there's another meeting. So there could be no, there could be yes. So every day or every other day, I'm calling for last, 1-2 weeks. But every time I heard one thing, that he is very positive, he would always like to come here. And I am extremely thankful that he has actually sidelined and reshuffled some of his very, very important meeting to make way for this particular gathering. And I request and a big round of applause for him. Thank you very much. And it was a real apt occasion that it's 100 years of the university where he has to be there. It's not that we can have this kind of function without him. And then it is also a sort of a blessing for a vice chancellor when a department is having a 75-year function. And on this particular point, I would like to express my gratitude to our previous head of the department who actually encouraged us, okay, you can have this kind of difficult time, but still you can have a physical conference. Professor Umesh Rai had been very instrumental in guiding us and actually showing us the way exactly how to do a conference in a university setup. And the day actually I put up the proposal, it was in April, the chair actually changed. And now we have another new person in the chair as a head, and she actually not knowing anything what has happened in the background too much. And then she open-heartedly and blindly followed and kept on signing those whatever request I had sent. I am extremely thankful to Mr. Veena Chakravarti for extending wholehearted support for this conference. And of recently, we have been, this conference is, if you are running alone, of course, you can certainly reach to the goal, but you cannot enjoy it. So we always found that there are very important sister institutions, particularly we have a sister institution, Ambedkar Center for Biomedical Research, and then we have Miranda House, we have Institute of Nuclear Medicine, which is of the DRDO. These all three institutions have been exceptional and phenomenal, support. Wherever and wherever I asked for support from these partnering institutions, they just came forward, whether it was accommodation, whether it was the scientific content. I think they all were there. And one of the most important, partnering institution is Ambedkar Center for Biomedical Research. So Professor Daman Saluja, again, she came forward and she helped. And we are so fortunate that she has now been heading our research council also. And this whole conference, if you actually look into it, this would not have been possible at all without the contribution of Professor Bharat B. Agarwal, because he is the one who conceptualized the conference on nutraceuticals, and we have already gone into 4 versions, and I am very much hopeful with your all support and your active participation, this 5th version will be equally, or maybe if God's grace, it will be better. Of course, the Ajay will start minding it, because the 3 of them he actually organized. Okay, so and behind this whole society, the Society for Nutraceutical Research, the main drivers are Ajay, and then strong support. Ajay is a very big person, and he's a very busy person also. He doesn't listen to me, but the person who always listens to me, whether it's a day or night, it's a Subhash, and he's from the same city. Please, Subhash Gupta. He has, for last 1 year or so, he has gone to Guwahati, and now he's grooming an institute called, it's a very prestigious institute. It's the All India Institute of Medical Sciences, and it's a Guwahati's institute. So he has got a head position there. But despite all the busy schedules, he was actually taking my calls and my requests what to do and how to do. And then Srijit is also there. And that was the society part. And then my whole department, I cannot keep, taking all the names. Everybody and anybody and everybody in my department, they really helped me in designing and drafting. And the most important person, which actually pulled me out whenever I was drowning, is Anju. Anjudi has been really instrumental to provide me a model as well as all SOS supports. So, and I would, a lot of things I can speak. I think I'll take your another half an hour or 1 hour when we are going into validatory functions. So please come prepared, take nice nap, and I'll be actually giving a very long thanks, lecture. Please don't mind at that time. But with this, we have a very limited time of our Vice Chancellor, sir. So with this, I will wind up and I'll say thank you very much, and please enjoy whatever time we have together. Thank you very much.
Thank you very much, sir. Now I request everyone to stand up for the national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविण उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्छल जलति तरंगा तब शुभ नामे जागे तब शुभ आशीष मांगे गाए सब जय गाथा जन गण मन कलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे जय।
Present conference will serve as a catalyst to start year-long celebration in the campus and dedicate ourselves toward goals of furtherance of higher education nationally and globally. We hope you will have an insightful experience throughout.
We have short few announcements. अभी उनका कर रहे हैं क्या कर रहे हैं?
So Vanika Gupta and session coordinators.
अभी session. The next session will start in 3 to 5 minutes, but I have some announcements to make. Please, for you, I think we have requested the university for a very important permission, and the permission is to visit Vice Regal Lodge. And there is another place, very monumental place in Vice Regal Lodge, which is Shahid Bhagat Singh's mark. And I think Professor Ratnabali ji has been very, very instrumental in getting this approval. And we are organizing a heritage walk at 2 different times. One immediately within the lunch, you will say it's not after the lunch, within the lunch and within the tea period. And we have kept a paper outside so you can mark your name so that we will assemble 5 minutes before that walk. And it is a 500 meter, 200 meter going, 200 meters coming, and then 100 meters within that particular area. It's a very short visit, but you can actually participate in that visit, and you can actually pay homage, and you can find how important this place is. This place, what you will see as a Vice Regal Lodge, was a major power center for around 100 years, where 5 viceroys of India had actually resided. And under the basement, in the basement, there is a dungeon that was kept for house arrest. So our Shahid Bhagat Singh ji, he was the one who was kept under trial in this particular dungeon, and we have created a monument. The university has created a monument there, and this is, and there is an archive. So anybody who is interested, there is no need of a car, there is no need of a vehicle unless there is a problem of walking. So you can actually walk through, you will pass through a cavalry lane, and then there is a garden. So you can go through that cavalry lane. You might have to sacrifice your part of lunch or deliberations during lunch or a part of your tea so that you can actually enjoy that place also. So please register yourself. We have kept a counter outside near the staircase so you can put your name and your phone number. Second thing, people who are the invited speakers, who have their outbound journey, so they can confirm their drop-off also to the airports and respective stations. So please check that also. So in the meanwhile, we are starting new session. It's a public talk by Professor Bhushan Patwardhan. I hope you will enjoy it. So we are reorganizing it. We will need 2 more minutes. Okay. इसको ठीक कर लो.
सर आपने बोल दिया तो
I request Dr. Vanika Gupta and Dr. Shashank Mourya to take over the session.
Actually, I put those things happening in front of you.
Yeah. Okay. It is my pleasure to welcome and introduce the chairpersons for the next session. So the Professor Reena Chakrabarti and Professor Kalpana Pai. Professor Kalpana Pai is from the Center for Advanced Studies at Savitribai Phule University in Pune. Professor Pai did her PhD in tumor immunology from Banaras Hindu University. She has held several visiting positions at reputed national and international universities. Professor Pai has made significant contributions in the area of biodiversity and immunoparasitology. I request Professor Pai to please come and start the session.
Namaskar to all the dignitaries, academicians, and dear students. So it's my great privilege and immense pleasure to tell you about the accolades and achievements of Professor Bhushan Patwardhanji, who is the chief guest of this occasion of INCD 2022. So he is the distinguished professor, Department of Health Sciences, Savitribai Phule, Pune University, and he is a national research professor at the Ayush facility in the University of Pune. He is chairman, executive council, National Assessment and Accreditation Council, that is popularly known as NAC, Bangalore. And his area of interest is evidence-based Ayurveda, ethnopharmacology, drug discovery and development, and integrative medicine. His brief bio sketches, he is PhD from University of Pune, formerly it was called as University of Pune, and his specialization was biochemistry. He was a research scientist in leading pharma companies like BioVed, Litaka, ISM. He is former vice chairman of UGC. He is former chairman of Indian Council of Social Science Research. He is member of several academic research and policy committees such as Niti Aayog, Planning Commission, Lancet Citizens Commission, National Board of Examination, and he is also into the Ayush India lead for the Global Center for Traditional Medicine at WHO. He is chairman, Interdisciplinary R&D Task Force on COVID-19. He is founder, editor-in-chief, Journal of Ayurveda and Integrative Medicine published by Elsevier. He holds 8 Indian and 2 US patents, and he has about 175 plus scientific publication to his credit. And I'll add a little bit more that he's very vocal on the sanctity of publication, scientific publication, and suppression of predatory journals by being chairman of CARE of UGC, that is the Consortium for Academic Research and Ethics. So he is one of the seminal members of in our country in making certain guidelines for publication, and it is especially for you all students and budding faculty members. And also I would like to stress upon the fact that he is one of the person who has brought scientific face to our traditional science of Ayurveda, which is based on philosophy and the practice. But he is the one who has, to the international community, he has brought the scientific face to it. So there are many more accolades, but I will stop here now, and I will request sir to please take over the stage and tell us about his journey into the science.
Thank you, Professor Pai. And thank you, students. I hope they will come. Professor Agarwal, as I mentioned, he is one of the inspirational personality in the field of nutraceuticals, and I could see many of his students also who have joined in various sessions. I had sent up my presentation. Is it there on the computer? It can be opened. Should I come there?
If it is all right.
यहाँ तो दिखना चाहिए.
Just share the second screen actually. ठीक है.
Yeah, thank you. Thank you. I'll skip the first part, which is actually my presentation. I'll going to talk about just a quick introduction to our school at Pune, what we do. Then I'll talk about nutrition, then immunity and inflammation, and then cancer, COVID-19, which is our own work. Just few glimpses of that. So at Pune, the Complementary and Integrative Health Center actually is a center of excellence supported by Ministry of Ayush. Our main activities include policy analysis, advocacy, and translation. We have several interdisciplinary scientific research projects. We also undertake teaching and training, and we have developed a collaborative consortia. The whole journey is from 1986 to 2020. I'm not going to go in details of that, but if you see, we have in silico activity in which network pharmacology is one of our current activity. Bioanalyticals, in which we are working on pharmacokinetics of some of the Ayurvedic plants. We also run a public health program, MPH, in which traditional medicine is one component. So these are several activities. We have also Patanjali Yoga Chair. Coming to the first part, nutrition, because this is about nutraceuticals, I thought of starting with nutrition. You know, nutrition, as we know, it is one of the major determinant of health. We say that there are 4 determinants of health, like 4 pillars of a building. And if any one of them is weak, you need support system, and that support system is actually medicine or medical care, which we say that healthcare. You know, we use health and medicine as well as healthcare and medical care as synonyms, which they are not. Health is determined primarily by 4. One of them is nutrition, second is environment, third is genetics, and fourth is lifestyle. Other than genetics, to some extent, you know, all 3 are in our hands. So we decide whether to go on a healthy path or unhealthy path. Nutrition, we are focusing today only on nutrition because the seminar is on nutraceuticals. But I just wanted to tell you that this whole decade UN has announced as an action on nutrition decade, as well as the Poshan Abhiyan, which Prime Minister has launched, is also a major nutrition initiative in our country. October 16, which is just coming up, is World Food Day. And Ministry of Ayush has come out with an innovative category under FSSAI called Ayush Ahar. Ayush Ahar is actually a modern nutrition or modern nutraceutical. Today we are living in a world when I want to raise these 2 questions, nutrition or nutraceuticals. And because Professor Agarwal is sitting here, you know, I wanted to make this point. Because today we are actually living in a medicalized society. We have started believing that by taking medicines we will get health. But health is too precious, you know, to be outsourced to doctors or to medicines. You know. Health is in our hand, and we have a great healing capacity within our own body. Our mind has a great capacity, you know, and we undermine all that when we actually outsource everything to somebody else, and we think that we'll go to somebody who is doctor, who will give us some medicine and we'll get health. No, we will get treatment for disease, not health. So like medicalization of the society, what we experienced during last couple of decades, now I am really fearing that we may enter into another problem of nutraceuticalization of society. So I want to raise this flag, you know. We talk about healthcare, but I want to ask this question, are we really doing healthcare? Or it is health scare. During COVID, you saw, we actually used to scare people, you know. You are going to do your diabetes, you are likely to get amputated, you are going to do this. So people get scared. And the moment they're scared, actually, the immunity is something which immediately is going to suffer, you know. So these are some of the first points I wanted to make. Second point I wanted to tell you that globally, if you go to any good journal, any good database, any medical database, or any reputed medical associations, and if you look at their recommendations and if you look at the meta-analysis results, you will find that there is a growing evidence to support that the importance of fresh and natural foods again. And I'm not going to go into all the details which you can see on the screen, but the systematic reviews and meta-analysis studies have not been able to provide convincing evidence that antioxidants, so-called, which are sold in the market. But mind you, there are excellent good antioxidants. I am not saying nutraceuticals are not required. Like I'm not saying medicines are required. Medicines are also required. Nutraceuticals are also required. But what is not required is overdoing it. The nutraceuticals are important, but let's not overdo it. Recent, just 2 months ago, if you look at JAMA, 2 consecutive issues, US PSTF, has recommended against use of beta carotene or vitamin E supplements for the prevention of cardiovascular diseases and cancer. This is JAMA just last month. Supplementation with vitamin D3 and omega-3 fatty acids doesn't reduce the risk of frailty. This is JAMA probably last month only. So there is a huge range of different kinds of products sold under health supplements, And if you look at this chart, you will see what different names people are giving. And it has become a huge market. My point is nutraceuticals are needed, but it needs to be supported by the work what Professor Agarwal has been doing. I don't mind turmeric, projecting there, because the whole lifetime work which he has done on curcumin is supporting that. So without science, today's nutraceuticals, if you look in the market, mindlessly combine something without even having safety studies. People are through aggressive marketing environments, we are being sold. So this is what I wanted to raise. Nutraceuticals are important, but let's not overdo it. And what we do has to be done in a very, very systematic and scientific manner. There is a way to do it, because there is thousands of years people are consuming something, and therefore safety is established. There is no question of safety when somebody is recommending me Triphala. If the Triphala is good, pure, I have no problem about safety. If it is adulterated, there is a problem, there could be a problem. But Ayurveda Ahar and the way of Ayurveda to look at nutrition is so exciting. And I must tell students that what Vice Chancellor was telling, you should come out with new ideas. Now, here are the ideas. Idea of dosha, dhatu, dravya, guna, rasa, virya, agni, ojas. These are not just terms in Ayurveda, Each one of them is a powerful research idea. One such idea, in my earlier career, I was using Ayurveda as a discovery engine, which I even now use it for preparing new drugs, But then I realized the real strength of Ayurveda is not only in medicine, the real strength of Ayurveda is in its concepts. So I started working on dosha. And I hypothesized in 2000, and we published that review, saying that Ayurveda is a customized medicine or personalized medicine. At that time, human genome was just emerging, so personalized medicine was not in mainstream. Pharmacogenomics also not on horizon, And at that time, we did a small study, because our group was working in rheumatoid arthritis, and we know rheumatoid arthritis and HLA relationship, we took HLA gene as one gene, because at that time, whole genome was not possible. I'm talking about 2000, 2002. And we showed that HLA-DRB1 gene polymorphism in a blind study, the distribution of allele was statistically different in Vat, Pitta and Kapha. For the first time, a scientific support in support of traditional concept like Tridosha or Prakruti came out. And now it has become a major multicentric study in which Ayurvedic biology, DST supported it under Dr. Valiathan. And now there is a report, if you can read in scientific report, that has received the highest citation, and the whole new discipline has emerged like Ayurgenomics. So this is a power of idea which is there in Ayurveda. I just wanted to mention. So the concept of Ayurveda to look at nutrition is different. They may not talk only about proximate principles and calorie. They will talk in different terms like rasa, guna, triguna, the sattvic, rajasic and tamasic diets. The effect, and we know if we eat tamasic diet, we get aggravated. Our mental frame changes. We know that. So these two charts I'm not going to explain again, but just to give you some idea to the students that how many innovative ideas are lying in our own traditional knowledge systems. These charts are systems chart prepared using systems nature biotechnology graphical notation technique, and these are forward backward linkages with color codes. You will see that logically from abstract concept like Panchamahabhut, to Ashtanga Ayurveda, everything is connected to each other in a logical manner. And if you take just one small component in red, what you are seeing here, it is Rasayana, which is one of the 8 branches of Ayurveda. It can be further expanded in equally complicated chart, which is just next to it, is Rasayana, systems Rasayana, So these are just glimpses of the richness of the knowledge systems, Just one systems Ayurveda chart is from 25,000 sutras representing 250 knowledge points, and each of the knowledge point can be expanded further. Based on this, we developed IUSoft, which is a software developed in collaboration with CDAC, which I'm not going to discuss now. Second part is immunity and inflammation. Now, Ayurveda, if you talk about, restoration of health using personalized approach is its key. And the focus is on physiological homeostasis. Bringing in the vitiated doshas to the normal, or from the Prakriti to Vikruti transition, bringing it back to Prakriti is the entire focus. And Rasayana are the ones, which we have used, and examples which I am going to give you are based on these Rasayana. These are actually tissue nourishment materials, and Rasayana need not mean only some physical material or medicine or some plants. There are Achara Rasayana also. That is lifestyle modification as Rasayana also has been described in Ayurveda. And most importantly, the immune homeostasis, not just homeostasis, but immune homeostasis and TH1, TH2 balance. So our work has been mostly in this area, of which I will just give few examples. These are the plants we have studied in our laboratories in various models, asparagus racemosus or shatavari, Tinospora cardifolia, or giloi, or guduchi, Vidania somnifera, ashwagandha, curcuma longa. I need not talk about it when turmeric man is sitting in front of me. Embolica officinalis or amalaki, and Glycyrrhiza glabra or yashti madhu, or madhu yashti, whatever you would like to call it as. The immunomodulation, there is sufficient scientific evidence that exists, and on the extreme right side you will see some of our papers have been cited there. So I'm not, because this is a public lecture, I'm not going to go into data as such. I'm just going to give glimpses. I know that the academic dean is also sitting here. She is a professor of law. So I need to talk something which all can understand, But the immune activation, cell proliferation, and marker modulation, these are the 3 important areas which we have studied, and we have shown that some of these plants actually, there has been upregulation of IL-2, IFN gamma, IL-4 and GM-CSF4. These are all related to inflammation. So immune modulation is further connected to immune inflammation. If you look at cancer and COVID, I'm going to give data or some glimpses of our work in these 2 areas. But if you look at both these, one may be communicable, one may be non-communicable or chronic, if you can say that, representing that, because the theme of this conference is chronic diseases. Both of them have one common path, and that is immune inflammation. And the work what Dr. Bharat Agarwal has done as researcher of TNF, is actually that led to his further research on turmeric or curcuma, and I was telling him, sitting beside him, his classic figure in which he has shown how one molecule can modulate so many different targets. That red figure which was shown in the earlier session, if you remember, that's a classic figure. Next part is about cancer adjuvant. These are actually our studies, but I'm not going to discuss the data. I'll tell you what we have done. We have shown that the immunity, chemoprotection, and quality of life, these 3 things we have said, because in cancer, when you talk about what is happening to, for example, NK cell activity, that is one of the research question, what is happening to cytotoxicity, what is really happening to the general inflammation, and then how do you prevent myelosuppression, which is one of the major side effect of most of the anti-cancer drugs. How do you really counter that without compromising the anti-cancer activity? Because if your drug is also modulating that, then it's of no use. and then ultimately reduction on various other parameters, it could be pain, fatigue, appetite. So we wanted to study all that. So what we did, we took one set of rasayana drugs in which ashwagandha and shatavari and guduchi, these three drugs were taken, and we mapped them against the backdrop of what we call it as hallmarks, cancer hallmarks. This is a classical paper. You may find cancer hallmark. There are 10 cancer hallmark, and for each of the cancer hallmark, we have found out that which mechanism and which plant is acting, which possibly can be useful for countering the cancer management, cancer treatment, primarily as an adjuvant. So we have this 20 days protocol taking experimental animals, and this was a completely controlled study in which we mimic chemotherapy. We actually mimic chemotherapy. In chemotherapy, paclitaxel is given, so we modify and we standardize the dose of paclitaxel to come out with an optimum level of desired myelosuppression, and by that way, on one we gave treatment as well. We developed a new kind of a model based on morbidity analysis, in which we scored rather the extreme tiredness or fatigue and peripheral neuropathy, numbness, weakness, hair loss, painful muscle and joints. These are all the side effects when you take anti-cancer drugs. These are some of the side effects which you face, and we actually could observe them, these signs in animals, and we marked them, and we created a score system for that purpose. In addition, we also could see that what is the picture of leukopenia, what is the picture of neutropenia, because these are the immediate side effects when you give paclitaxel, it is coming up. And if you see the first, which is the control, second, when you give paclitaxel, you will see that there is sudden suppression of leukocyte count, and when you give these drugs, you will see that the original levels are maintained. So this is clearly showing that in our model that ashwagandha could actually protect myelosuppression without compromising the anti-cancer activity of paclitaxel, which is expected to be done. So in short, the results showed that there was decrease in activity and behavior after paclitaxel administration. There was increase in hair loss and hardening, and there was change of the hunch postures. So all this, when we actually gave treatment in the treatment groups, were substantially improved by way of course. So this is this cytokine profile. We did the entire cytokine panel, or 20 cytokines we did, and you will see that the red is showing the down regulation, the green is showing the up regulation, and overall, if you see that ashwagandha and shatavari both modulate PTX-induced alter cytokine levels, in a positive direction, bringing benefits. So in summary, these experiments showed that traditional medicines, including ashwagandha and shatavari, prevent paclitaxel-induced myelosuppression. By prevention of myelosuppression is countering leukopenia and neutropenia and preventing the other side effects. There is also downregulation of oncogenesis, morbidity associated with cytokines, and there is upregulation of hemopoietic cytokines to achieve immune homeostasis. That is normalization of cytokines. And then we thought that similar activity of ashwagandha, which is actually immune modulating activity, which is Th1, Th2 balancing activity, may be useful in some other conditions as well. And here the issue of COVID-19 came in. COVID-19, what is important? First is viral load prevention. The entry itself, when virus is trying to enter, can you do something? So we did some work in that area. We also used the nasal route, There is Anu Tail, is a preparation, Ayurvedic preparation, and Professor Madhu Dikshit from Thisti did these studies using hamster models, and actually she has shown that by applying this Anu Tail, there was antiviral activity, and she has published also that the paper we have just finished here. Now, then immune homeostasis, balancing immune, because there is cytokine storm. How do you really deal with that? And the third is organ protection. In advanced cases, there could be organ damage. So these three things we wanted to look at. And then a probable role of ashwagandha in COVID-19. If you really want to know more about it, we published this paper in one in Current Science and other is in JACM. Both these papers have received very, very high citations. And we could clearly show that in COVID-19, and there was a control study. In one group, we took ashwagandha. In other group, we took hydroxychloroquine. Because hydroxychloroquine at that time, if you remember, in the initial days, for high-risk workers, hydroxychloroquine was recommended by ICMR. And we actually argued that when we have a safer option for hydroxychloroquine, why you are not including that? But it was not heard. And we had data also to show, because in rheumatoid arthritis, where we have shown that ashwagandha was found to be much better than hydroxychloroquine, in a controlled clinical trial, randomized controlled clinical trial. But still, they were not ready. So we undertook an independent study. This was multicentric study. This work is also published in a multicentric randomized controlled clinical trial done not by Ayurvedic practitioners, but in modern hospitals. We have shown that ashwagandha is a better drug for prevention of COVID-19 than hydroxychloroquine. During last 2.5 years, Ministry of Ayush has done several studies. I am chairing that task force on Ayush or COVID-19, and these centers include almost 126 studies, 130,000 participants in these studies, and 150 sites across India, not across India. Some of the sites are also outside India, So this is a huge study. Many of these studies have been published. Some of the drugs which we have studied, not only ashwagandha, but in collaboration with CSIR, we undertook 3 major studies, randomized controlled clinical trials. One on ashwagandha, which was for prevention. Second on Yashti Madhu, that was for adjuvant treatment. So there was standard treatment. And in addition to standard treatment, Yashti Madhu was given. In addition to standard treatment, Giloy was given. So these were the groups. And we could show that, and Ayush 64. Ayush 64 was a medicine which was developed for treatment of malaria. And this we repurposed it for the use of in COVID-19 situation. And we could show that Ayush 64 actually reduced the number of days in hospitalization, which itself was a very big requirement or achievement during our need for that time, because hospitals were getting overflow. So all these studies show us that if a robust protocol is developed, that is the first and most important thing. Most of these protocols we published in advance, So the credibility of the studies also can be verified and checked. This is all in public domain. So these are not the studies like Me Too studies wherein you just give some drugs to patients and they say percentage wise they are feeling good or better. These are systematically planned studies in which we have shown that prevention, ashwagandha is better than hydroxychloroquine. As an adjunct treatment, Ayush 64 and Giloy have much better effect in addition to standard of care. And third is post-COVID management also. Delhi police, 30,000 Delhi police were given Giloy and a kit, which is Ayur Raksha kit. And the study of that recently has been published in Frontiers in Public Health. You can refer to that study in which we have shown that most of the people were also surprised, those Delhi police who consumed this treatment did not get COVID during that study period of time. So something definitely has been demonstrated today through Ministry of Ayush, raising hopes raising interest of the scientists also. And I personally feel that I'm not an Ayurvedic physician. I am biochemist, I started looking at Ayurveda earlier from drug perspective. Then I realized that there is much more than drug there. And in terms of nutraceuticals, if you are able to really look at Ayurveda, you will get not only one turmeric which Dr. Bharat Agarwal has given. He has just given us one example, but there are hundreds of such sitting there, waiting for other Bharat Agarwals to come in and take them to the next level. And I hope that some of the students really and teachers take this as a challenge and do it. This is our ongoing study. This is again a multi-centric study. Based on our earlier studies with Serum Institute of India on vaccines, at that time we used DPT vaccine, but we showed that if you give ashwagandha orally alongside with DPT vaccine, the immunogenicity was increased of DPT. And we wanted to test this hypothesis on COVID vaccination also. So this study was planned. This is already, we have completed the recruitment of participants, 1,200, and this is multicentric studies. 7 collaborating institutes are participating in it, which is led by University of Pune. The protocols are approved and everything. And what we are giving is persons before taking COVID, when their date is known to the COVID center, we recruit them. We studied their entire body chemistry and anthropology and everything, and then we start treatment. And this is going to be a long-term study. So this is a 6-month study. What happens immunogenicity, and at 2 levels, we will be drawing blood and we'll be looking at various markers in it. This is going to be a very interesting study. Protocol of this study has already been published in Frontiers in Medicine. Those who are interested can look at that protocol. And this study, we expect that if there is even a moderate success in this study, India will have a tremendous opportunity in the field of immunization and vaccination for improving, because this is not only COVID vaccine requirement. BCG requires it. Every vaccine now will require this kind of. So we are trying to use a different innovative way by not putting something in vaccine as adjuvant, which also we have done, by the way. The data I have not shown here, which we have done in collaboration with NCI and Memorial Sloan Kettering. Memorial Sloan Kettering actually has worked on our material as vaccine adjuvant, which cancer vaccine developed by them. And they have shown excellent results in comparison with the other vaccine adjuvant which they have been using so far. I will end here by telling you that Ayush Ministry took the advantage of the adversity, because our Prime Minister also appealed to us, this is adversity, but how can you convert it into advantage? So we converted that into advantage and undertook an example of studies which can be done on Ayurvedic medicines without losing the rigor of science, without losing the quality of medicines, and without making any tall claims how these studies can be undertaken. What we could do in 2 years in COVID-19, several other chronic diseases are waiting for us to do these kinds of studies. And I am so happy that all of you have gathered together here to discuss nutraceuticals. So I would say that nutraceuticals, don't imagine that nutraceuticals are only something which you get in the market, because that is not the correct representation of nutraceutical. The correct representation of nutraceutical is what Dr. Bharat Agarwal has done by taking turmeric as an example, So go on that path and not just mixing something and putting something in the market, calling it as antioxidant, or putting so many different claims without any kind of a validation. With this, looking into all the work which is happening in India, and also looking into the potential of traditional medicine, and also knowing the growing limitations of current therapies, the World Health Organization has decided to establish Global Center for Traditional Medicine in India. The center has already been inaugurated by the Prime Minister and the Director General of WHO. This center will be established in Jamnagar, where India's first Ayurvedic university is present, and it will look into various aspects of scientific research in traditional medicine, and the evidence and learning, data and analysis, the sustainability and equity, and the innovation and technology. These will be the main streams which this center will focus on. With this, I would really like to thank all of you, especially all the organizers, Delhi University, all the delegates, and I also want to thank my own colleagues, faculty colleagues, those who have worked with me. Whatever I have presented is not only by me, but my students, collaborators and funders. I would like to thank all of them. Thank you very much. Namaskar. Jai Hind.
Thank you so much for such a wonderful and enlightening lecture. I think it will be an inspiration for the budding scientists to take over, that indeed, our plethora of Ayurvedic treasure is there to open, and for food as, for health. So thank you so much, Professor Patwardhan ji. Organizers, please.
On behalf of the conference organizers, I would like to thank our chairpersons, Professor Pai and Professor Chakrabarti. I request Professor Namita Agrawal to please come and felicitate the chairpersons with memento. Thank you. So we'll break for lunch now, and we'll see you soon for the next session. Thank you. So we are running behind the schedule. So I think it will be a small lunch break, and please do find time to go and see the posters upstairs. Thank you.
डिवाइड स्क्रीन हो रही है यार आधे ब्लॉक में आधे ब्लॉक स्क्रीन डिवाइड हो रही है प्रेस करा दो ऊपर लैपटॉप पे खाली हो गया थोड़ा कॉल हो हम है थोड़ा सा भीड़ चलती रहेगी आप ना सोचो अब इकट्ठे खत्म हो जाएगा चलो
ऋषिमा, यहाँ पर S1A है। ओके, हमारे पास रजनीकांत सर का है, सिद्धांत महन का है और रविंदर जी सिंह का मैं डाल दे रहा हूँ। हाँ, और एक और यनी शेट्टी हैं, उनका बचा हुआ, उनका नहीं मिला। एक अनाउंसमेंट तो करनी पड़ेगी।
मैम ये वाले इधर ही लगेंगे ना, ये सब इधर ही लगेंगे ना।
ये रविंदर जी सिंह चाहिए, आपको बुक देंगे कहीं ना पास। नेहा, वो बुक पास करेगी क्या? थैंक यू। ये सेशन 1 है, तो हमारे पास है प्रोफेसर रजनीकांत का, ठीक है, ये छुट्टी का आना अभी बाकी है, सिद्धार्थ महन, ओके, और फिर उसके बाद नरेंद्र सर हैं और इल्लोरा सेन, ठीक है। तो नरेंद्र सर और मेहन का बाकी है, और एक और मैं फोल्डर बना दे रहा हूँ जो अगला सेशन है। अगला सेशन कौन सा है मैं देख लेता हूँ। ये वाली है ना, ये सेशन 2A है, ठीक है, S2A, ओके, ये मिल जाएगा ना आपको आराम से।
अच्छा, ये मुझे करना है
अगर इन केस
अच्छा तो दिखाओ फिर से। क्या दिखाया
एक मिनट,
फर्स्ट क्या दिखाया था?
एक मिनट मैं दिखाता हूँ।
पहले वाला फोल्डर भी दिखा दो।
वो भी मैं दिखा
पर मुझे नहीं है आइडिया यार, इससे मैं कैसे क्या करूँ।
कुछ नहीं, बस आपको खोल के स्टार्ट कर देना है।
क्या स्टार्ट करना है दिखाओ।
उनका सेशन एक सेकंड मैं पहले बस इनका कॉपी कर लूँ ना, उसके बाद दिखाता हूँ।
यार वो नहीं सोता बैठे उसको बुलाते हैं, उसको सिखाते हैं, ठीक है बुला लेना। श्रेयता श्रेयता श्रेयता,
देखिए
इसको बुलाना है श्रेयता को।
ये सेशन 2A है जो 4 बजे से शुरू होगा। ये सेशन 1 है, मेरे पे मत डालो यार, ठीक है। ये सेशन 1A है, ओके, ये कहाँ गया, सेशन 1A और ये सेशन 2A, ठीक है, चलेगा ना। 1A मतलब अभी जो शुरू होने वाला है, और 1A के सारे स्पीकर्स यहाँ पर हैं, 2 अभी आने बाकी हैं 5 में से, ओके। स्टार्ट ऐसे करना है, ये करना है, इसको ओपन करना है।
तो तुमने ओपन ही रखे ना।
हाँ,
आगे अपना ओपन कर लेंगे, है ना।
ये चलो वो नहीं हुआ,
तुम इसको इसी फोल्डर को ओपन रखते हो।
हाँ, नहीं सर, ऑनलाइन नहीं जा रहा हूँ सर, यूट्यूब पे अभी सब ऑफ ब्लैंक है। यूट्यूब पे तो वो जा रहा होगा, जो नॉर्मल पिक्चर जाते हैं ना वो जा रहे होंगे। हाँ, एक रन करता रहता है INCD का पोर्टल।
और इसको तुम ये फोल्डर ओपन ही रखना ना अपने आप।
ये फोल्डर होते हैं। क्या कर रहा है? कुछ भी डिस्प्ले नहीं हो रहा है। क्या अगले सेक्शन के लिए जो फोल्डर रेडी कर? हाँ। नहीं लैपटॉप ठीक है। छोड़ दूंगा तो फिर कैसे होगा? वो फिर बहुत अनएक्सपेक्टेड हो ठीक है। और अभी मुझे दो स्पीकर येमी शेट्टी हैं, नरेंद्र हैं, ठीक है, उनका चाहिए।
हॉल बी में है स्वाति मैम कोऑर्डिनेट कर रहे हैं।
Yeah, I got it. Yes, I believe so. I'll show you. So please.
Yes.
This is fine? Yeah, thank you, sir. Yours is third, right? हाँ पूर्वा, हाँ बोलना।
If you're a Texas homeowner, 2022 is your last chance to go solar. In December of 2020, Congress passed an extension to the solar investment credit, which pays homeowners an average of $9,880 to install solar panels. See how much you can be paid for going solar by taking the Go Solar quiz below.